chemicals. So in the era of CRISPR-Cas, why tilling is still important? I will be talking about CRISPR-Cas, but the relevance is not going away. Simply because each of these line are having millions of mutation, we have NGS data, we have bioinformatics software, we can quickly go through all the results in a very limited time, and we can identify multiple alleles. See, there are what I said before, that there are 30,000 genes in rice, 5,000 we know, but that is not enough. Even within those genes, one nucleotide change can create a new spectrum of allele. So there are millions and millions of alleles which are. So it is just not that I know the function of the gene and gene gives enhanced grain yield. So that is my endpoint. I can change each of this nucleotide just like a typewriter. I can change each word and the meaning will keep on changing. Even though all of this, say a gene has 1,000 base pair. Now if I make 1,000 different changes, they may give 1,000 phenotype. So just knowing gene function is not the end of the world. Creating allelic variation and each attributing each variation to the same phenotype but enhanced phenotype is critical. So there is a lot to be discovered among during the coming years. Tilling, why tilling matters? It works in recalcitrant genotypes. So if you are working with CRISPR-Cas, you at present here it works in all the genotype. You don't worry whether we can transform the genotype or not. It is scalable and cost effective because say you have 1,000 lines, you mutate it once, and then you can population that data for several years and decades to come. So it is a universal resource. Next. So from the national point of interest and for the private sector, what are the traits of interest for us? And since we work for farmers, so we develop traits which the farmer needs. So what the farmer needs in general? Farmer needs enhanced yield. Farmer needs disease resistance. They need products which give abiotic stress tolerance. They need a very perfect plant architecture. For example, it could be mechanized harvesting in any crop. Flowering time is very important. If you can have early flowering and then there is drought, then you can escape the drought. So those things are needed because you know the rainfall pattern has completely changed. So these are the need. Then fruit or seed ripening, that is an important parameter. And then herbicide resistance. So all these are the traits which farmer needs. So for commercial aspect, what for us? For us, it could be a hybrid seed production system. It could be traits which can be used in food processing industry. It could be like different varieties of tomato. It could be biomass which we can use for say electricity production or bioethanol production. So this is from the commercial aspect. And what as a consumer, what we need? We need reduce toxin from fruit and seeds. We need increased nutritional value. And we also need, if there is any allergen, say for groundnut, there is, or peach or apricot, there are many food allergens. Can we reduce it? So consumer needs these traits. So having said a lot theoretically, where is the proof that we have innovation? Acid in rice, high amine-free and improved oil content and protein. You have reduced lignin in bajra. Then you have extended shelf life and potyvirus resistance in tomato. Then amylose-free potato. These are the different products which are available in the market. Then you have low synapine content in mustard, and then you have shelf life in melons. You have colored cauliflower. So they all come from the mutation breeding. And here only I'm just going to talk about a few examples as I mentioned in the previous slide. So what it means by high amylose content. So wheat with high amylose than the normal wheat. It produces more resistant starch, low glycemic response, mainly from this gene, SBE2 gene, through the EMS mutagenesis. Similarly, you can also recreate it through tilling. It has low amylose, high resistant starch, first non-transgenic varieties which are released in wheat. Next. And then oil quality. Oil is a very important for us because every year we are spending 1,40,000 crore rupees, just lakh crore rupees to import oil. We have a lot of dependency on other countries, European countries, Russia, Ukraine for cooking oil. Here also you can modify the fatty acid profile, you can improve shelf life stability, you can have specific variants which can be used for SAD, and then that will be for industry desaturates. This will is a useful acid for human health. So oil quality is improved by for human consumption. Next. So all these varieties which are developed through mutation, mutagenesis, they all have exemption under European rule for in the organic system or the organic produce. They are accepted in the organic system, and there is already a consumer trust which is built up around this mutagenized crop, and therefore we expect that gene editing technology will be equally accepted because you are only doing a selective change rather than changing the entire genome. Till now, consumers are wholly satisfied and wholly trust those mutagenized crops where intended, unintended, all mutations are there. So now when we are producing something through editing and we are just changing one nucleotide while keeping the rest of the system alive, I hope that it will gain more consumer trust. Next. So now I will be talking about from induced mutagenesis, first part of the talk, to genome editing, right? Evolution for crop improvement. So induced mutagenesis enabled non-transgenic trait discovery for decades, more than 100 years. It generated valuable alleles for diversity, or yield, quality, and stress tolerance. Genome editing enables targeted trait modification and faster deployment, reduced linkage drag compared to conventional mutation screening. Change, targeted change. Mutagenesis and editing are complementary approaches. So all the thing I have told about mutagenesis, now you are doing a very targeted change. And mutagenesis remains important for novel allelic discovery and complex trait exploration, whether you do through direct mutagenesis or through genome editing. So in this slide, I am saying that how this genome editing came into effect. It is a modified prokaryotic immune system. So I will give one small story about the genome editing, how it came into being for the young researchers who are pursuing your master's and PhD. See, bacteria and virus, they are always at war against each other. Virus needs bacteria to replicate, and bacteria needs to avoid the virus because they will get killed eventually. So basically, whenever the virus attacks bacteria, it copies a fragment of the DNA and it keeps that DNA in its library. So second time the bacteria will come and attack the virus will come and attack the bacteria. Bacteria now recognizes that virus. So it is an immune system of the bacteria. So what happened is in 1980s, a PhD researcher, while sequencing, because that time the sequencing technology was very crude, all these recent developments were not there. So she identified some unusual sequences in the genome of bacteria, but that's it. She submitted the thesis, got her thesis. Someone else in Spain found similar sequence in archaea, and he cross-referenced that with this PhD thesis. And then he also realized that this trend is there in all the higher organisms also. So we knew that CRISPR-Cas is an innate immune system. And it took a long year with several of researchers spending time and energy till they realized that if we can give a guide RNA, then we can direct this nuclease to that portion of the genome where we want to edit. And not until, so this discovery was done by Jennifer Doudna, and Emmeline Charpentier came to her lab to do some research. Now, Emmeline Charpentier, she changed 5 labs in 5 years, and she's a very dedicated researcher. So when she joined Doudna's lab, she was trying to edit the system, but they could not edit the system because there was this single guide, trans guide, and there was this protein, but there was no editing. So why she got the Nobel Prize? Because she was involved. She realized that you also need a trans guide RNA. So you make a basic loop. Without that loop, there can be no editing, and that is why the Nobel Prize was given to both of them. So that is how the editing system came into form. And you can just change any code. So now there is base editing, prime editing. You can now do specific changes. When CRISPR-Cas came in 2012, these were all random changes in that particular segment. Or you could give a donor DNA so that you can do a specific change. But with prime editors, now you can do a one single base pair change where you intend to do the change, not any other change. So it is saying it just works. So genome editing in global commercial breeding is getting rapidly adopted across the seed and biotech companies. Focus area is similar as same as I told through tilling. It accelerates breeding cycle when integrated with markers and speed breeding. So these are all the basket of technology. These are tools. So you can use as many tools to compress your timelines. So without this, can of years? Can you wait for thousands of years? You need the solution now. And now, then CRISPR is the best tool. Because now you know so much information is there. You can change one nucleotide, which can, whether it is in plants or whether it is in disease therapies, where one nucleotide change can change between a cancerous state and a normal state. You can do very precise changes. The patient is not going to be here forever if you go through the normal process. So all the gene therapies and all the CRISPR use breeding can take place. We can get there much faster. Next. So what is the regulatory scenario? So as long as you are in SDN1, so there are three categories. One, there is only small nucleotide change. Now, when I mean small, it could be any number of nucleotide change, but it is natural. There is no DNA template. Then you have the second type where you have to give it a small template so that after the cut is made, the cell is able to copy that segment. That is type 2. And type 3 is transgenic, where you want to change the entire allele. Suppose so, as far as India is concerned, SDN1, this we can, it is free to go. So you have to go to your institutional biosafety committee and submit a dossier. And why we need this? Because we want to make sure, see, the machinery used most of the time is with recombinant DNA. Of course, there are other techniques also like RNPs or the synthetic version of the guide RNA and the protein. There is no integration. But Government of India wants to make sure that there is no rDNA in the line. So they have made this some that these are the points you need to cover to make us and all of us sure that there is no genetic elements left there after the editing has happened unintentionally. So that's why we have something here. If you go for other SDN2, then it will go to the regulatory body and they will see it on a case-to-case basis. And if it is SDN3, it is regulated like GM crops. So mutation breeding in SDN1 is most acceptable. And similar trend is there in US, Japan. All the countries are now opening up because with the SDN1, because they have already been doing mutation breeding for thousands of years. Then what is the point to regulate it? We are already consuming it. Now we have better versions which can do that work in a very small time period, say within 6 months. Next. So having said that, we are in the era of artificial intelligence and agriculture is no exception. artificial intelligences will be used everywhere, including genome editing. We can designer crops. So you see how much progress we have made in last 100 years. We were doing all the random mutagenesis. We were tracking for the phenotype. We went from there to generate DNA data, mine for the specific allele. We went one step ahead in generating next data, terabytes of data through next generation sequencing, and we expedited the process. Now we have to go one step ahead, and we have to integrate artificial intelligence. So if I say this is an intelligent driven crop breeding system, basically this is the new for say 2026. And here it is like high throughput phenotyping analysis, then biological big data driven functional genomics, then AI based breeding combination and selection. We were using different model to select, okay, this combination will be better, but there will be billions of combinations. Just like the neurons in our brain, we have billions and maybe trillions of connections which can be made. Similarly, all the combinations can be tested. If we do it through any other model, it will take days and years to evaluate that data. But through artificial intelligence, we can speed it up. So how much the progress has gone ahead, and that is what I call the intelligence hybrid breed. So this is how AI is integrating. You generate the data, you make sense of it using AI-based algorithms, which are much superior than single models, just like a neuron-based connections. And here you select those combinations, and then you can make similar changes. You can recreate those alleles as predicted by AI software through genome editing, or you can screen the existing population because you already have the data whether those mutations are already present. Because if it is present, you can go ahead and bring that line. There is no need even to make a dossier or document. So mutagenesis, AI, and genome editing, this is convergence of these technologies for next generation crop improvement. What I want to emphasize in this presentation that, and I started from the history because then the students will be able to understand that how far we have come. What you know yesterday and what you know today, there is this linkage. So genome editing, it is the precise modification of genes controlling yield, stress tolerance and quality traits, rapid validation and trait stacking in breeding pipelines. And when you use big data and AI in what I call the intelligence hybrid breeding, you are integrating genomics, phenomics, environmental data, all that data and all the genomic information you have. The AI software is able to prioritize your mutations for gene editing. As I said, you can create millions and trillions of combinations, but among which to use. This will AI-driven prioritization of mutation will help you to prioritize what you want to do with your limited resources, and then you can edit the gene. So what the future of breeding will look like? Future of breeding will be a combination of mutagenesis through CRISPR or chemical physical mutagenesis or through natural variation. And by using AI prioritization, we can breed much faster because environment is changing at a faster rate than we can even act on. Before we know that the environment has changed, disease has occurred, everything around us changes. How do we act and adapt? This is through future breeding paradigm. Next. So future outlook towards predictive breeding. So all what I mentioned there, we are transitioning from what used to be only largely phenotype driven breeding to prediction driven breeding. We are using AI guided mutation and genome editing targets. We are integrating genomics, proteomics, phenomics, and environmental data all together. Otherwise, it would not be possible. We were acting in silos. Now we can integrate all that data, and with all the systems available, Amazon Cloud and all this, we can put large amount of data so everyone can use from everywhere. So you don't need to go to institute to do that. Everything can be done there. Faster response to climate variability and emerging stresses. You can react much faster, whether it is in crop or it is in human health. We can work much faster, and expansion of advanced genetic tools across crop. Future lies in integrating diversity, generation, precision editing, and intelligent data system. So this intelligent data system, which you all will be working, those who are just entering their masters and PhDs, this is what is going to be the future. Next. So if I specifically talk about CRISPR-Cas, each and every area it will touch on. So all the big data you can use not only for human gene therapy or agriculture, you can use to screen for drug ID. You can use for ecological control, viral gene disruption, pathogen gene disruption. You can fight against future pandemics whenever they come. You can do synthetic biology work. You can re-engineer the pathway to understand more about the genetic system. You can do programmable RNA editing. You can change the RNA. There is no need to do even the, we are working towards proteins. What if you change the RNA itself? So if a programmable RNA, which was unthinkable even now, this can be done through CRISPR-Cas in the future. Next. So what is there for the stakeholders, for all of us? There is higher acceptance of the intelligent breeding system. There is science communication is very critical. We have to remove misconception. And there is the bridging policy and innovation. Everything is coming under stakeholder perception. Next. So what is the commercial advantage? There is is a clean patent scape, and this particularly is for chemical mutagenesis that is free to operate in the breeding system and startup, because a lot of startups are coming out right now. Then they can more is more friendly for innovation. They don't need all the expensive instruments. They can with their limited funding they can move ahead. Next. So what are the convergence mutagenesis, as I said, with genome editing, NGS, AI-based phenomic platforms, speed breeding integration, all these things from genome, proteome, metabolome, NGS data, and all this, along with speed breeding integration, you can rapidly increase the development of varieties in the future. Next. So what are the final key takeaways from my presentation? Mutation is evolution accelerated. Mutation is happening all the time before we realize. And what I talked is mutation of the evolution accelerated. We are just accelerating to our changing time. Diversity is resilience. Our diversity of germplasm is decreasing. So by using all those tools, we can increase the diversity. And next green revolution needs young scientists. So scientists like you will be able to exploit AI-enabled green revolution in the future. Next. We have edited one on tilling and one on. So we have explained all the fundamental examples and all things. These books are already available, and CRISPR book has been highlighted in Mukesh Ambani's favorite, one of the favorite books in one of the podcasts. Next. So thank you very much. And what I want to say in the last few words, all science take inspiration from nature, but nowhere is this more true than in biology. This is where we spend. Thank you.
So please have a seat. And please, if anyone have any question, the session is open for discussion. Anyone from faculty as well? I think no one has any question. You have?
Sir, if you take on a gene from crop that you are consuming now, that is, suppose it is a drought resistant gene, and that gene will inserted in another crop. It is accepted by a regulatory authority.
See, if you are developing similar allele with any of this technology, you are not inserting it. You are rewriting the genetic code.
The both crops are different.
Yeah, but the gene is same. See, gene which attributes to yield, many genes are spread across crops. They are similar nucleotide. Some changes are there. So here we are not taking one gene from rice and inserting into wheat. That we are not doing. We are taking the information from rice and we are trying to create an allelic series in wheat. We are recreating the mutation. So we are not changing anything. It is the wheat genome and we are changing that word. Say low yielding, and we are now changing that low to high. So this is not transgenic. This is completely mutation breeding.
Hello. Sir, first of all, I would like to thank you for your nice presentation. You talked about the conventional methods of mutagenesis and then you ended with the recent one that is CRISPR-Cas editing. So just I would like to ask, in the era of CRISPR editing, so how that older methods are still sustaining, that is conventional mutagenesis, because nowadays everyone is talk about the fancy molecular biology technique like CRISPR. So I think you are working on that, so you will answer it better way.
See, tilling is still relevant today because, as I said, that say there are 1 million nucleotides, and changing one at a time will create a new allele. So, tilling will help us in understanding and undergoing the basic science too. And then for applied science, we can use the same information to create through gene editing. The other thing is in gene editing, if you want to every time create a new allele, you have to go through the same process. In mutation breeding, you have all you mutagenize only once, and you screen for that data. If you already have the mutation, then you don't need to do it by editing. If you don't have a something parallel, then you go through editing. So tilling is still relevant in CRISPR-Cas9.
Sir, again, one thing I would like to ask, as far as regulations are concerned in CRISPR technology, I think in India the stringency is very high. Even though we are having the permission of only changing few bases, one base, two base like that, R factor is there. So as the food demand is growing, population is growing, and I think India need to move ahead with the GM, even though in trade deal, the US is trying to send some
Bonds.
crops in India. So why we are not that accepting that fact and we are not preparing it for our generation?
So there I talked about consumer trust. Building consumer trust is essential. When the green revolution happened throughout the world, it came from public institutions. So there was a lot of trust. When it came to GM, then lot of misconception aroused, and primarily arised, and it all primarily came from the fact that private organizations were involved. So this is the difference. So we hope that gene editing now this is universal. It can be used by startup, government, industries. They all have a level playing field.
Okay, sir. Thank you. Thank you.
Do anyone have any other question? Yes, at the last.
Hello. If mutations are random, then how would you increases the useful probability of useful mutations?
No. See, mutation is random, that is true, but we have to screen for the useful mutation from the random mutation. Why they are random? Because this random mutation helps plants to survive. If you have a large mutation or you delete a gene entirely, plant will die. But all these random mutation are helping the plant to survive. And then we have to screen for that particular mutation, and that is possible today because of the all the next generation data capacity which we have. And as I said, in our times, in this era, we have AI driven intelligence breeding. So we can make a lot sense of the data than we could have made earlier. So now they say data is the new currency, data is the new power.
Thank you, sir.
So I also have one more question. Like Bhagat sir also talked about single or multiple base editing at the same time. In case of like sickle cell disease and there is one disease also CPS1 deficiency. So we uses CRISPR to just tackle these conditions.
Yes.
So what do you think about multiple base editing at a single time using CRISPR? So
Yes,
it is
that
possible
is
as of now?
yes. So now even using CRISPR, and that's what we are deliberating in our lab. You can. I was in Delhi last week for a conference, and they have the government is NIPGR. One PhD scholar showed that he has been able to edit 400 genes at one time. So multiplex editing has gone to that scale.
So, but currently, like there is one company known as a Colossal Bioscience, which works basically in the extinction process. Which like we had a discussion in classroom also on this point. This company just edited 14 genes, and in that 14 genes, they specifically like made 20 cuts for the de-extinction of one animal species known as a Canis dirus. So as of now, the like such big companies only proceed for like 20 edits and 30 edits as per the 14 bases or 14 genes. And you were talking about 400. So
400,
yep.
yes, that a PhD student has done. So you can understand that there is so much for people who are undertaking PhD studies or postdoc studies today than what was possible in my times. So that previously we used to say that everything changes in 10 years, then in 5 years, and today we say every year it changes. So evolution of all the technologies has also accelerated. So a lot of scope for you guys.
Okay, thank you.
Thank you.
Thank you very much.
Now I'll invite Dr. Peshwa Madam, the chairman of this session, for her concluding remarks. Peshwa Ma'am, please.
No, thank you, sir, for a very thought-provoking deliberation. So I conclude now. Harnessing induced mutagenesis stands as a powerful and proven strategy for next-generation crop management. In an era marked by climatic uncertainty, resource constraints, and rising food demand, induced mutagenesis offers a practical pathway to generate novel genetic variability and accelerate the development of resilient, high-performing crop varieties. When thoughtfully integrated with the modern tools such as genomics, high throughput phenotyping, and new plant breeding technologies, induced mutagenesis, along with the targeted genome editing by CRISPR-Cas9 technology and AI driven crop breeding, can significantly shorten the breeding cycles while maintaining public acceptance and the regulatory ease. Its strength lies in its versatility, supporting improvement in yield, stress tolerance, nutritional quality, and sustainability. Moving forward, the true impact will depend on interdisciplinary collaborations, precise screening platforms, and strong linkage between laboratory innovation and field level deployment. By combining the classical mutation breeding with cutting edge molecular insights, we can build robust climate smart cropping systems that meets the needs of both farmers and the consumers. The future of agriculture will belong to approaches that are innovative, inclusive, and scalable, and induced mutagenesis and targeted genome editing is poised to remain a cornerstone of this transformation. Thank you. I hand over the mic to Samuel, sir.
Thank you, ma'am. I'll now proceed for the vote of thanks. On behalf of the organizer, I extend my sincere gratitude to Dr. Bhattacharya sir for an enlightening and thought-provoking talk on harnessing induced mutagenesis for next generation crop management. Your insights into the strategic integration of induced mutagenesis with modern breeding technology have been truly inspiring and valuable for the scientific community. The depth of knowledge and clarity of presentation has greatly enriched our understanding of future ready crop. We deeply appreciate your time, expertise, and perspective that you have shared with all of us, sir. I would like to also extend my heartfelt thank to the chairman of our present technical session, Dr. Peshwa ma'am. She is one of the senior most faculty of Government Institute of Science. She has been serving here since more than 26 years, and having such an eminent person as a chairman for this session was we have greatly appreciate her presence and her time as well. Thank you, ma'am. So on behalf of this committee, organizing committee, I declare this first technical session is over. Thank you.
Thank you all for the smooth conduct of this keynote address. Now we are heading towards the technical session one, for which I invite our resource person, Dr. Khushbu Burel ma'am, to kindly come upon the stage. And also I request the chairman, Professor Petkar sir, and session coordinator, Taipei sir, to please come upon the stage. I request Director Sir to kindly welcome and felicitate our resource person, Dr. Khushbu. Also, I request Inder Sir to kindly welcome Inder Sir and Taipei Sir.
इसमें डाल के दे रहा हूँ मैं आपको कॉपी
I also request Inder Sir to welcome Taipei Sir. Thank you, sir. With this, I now request Professor Petkar sir to introduce Dr. Khushbu to all of us and take charge of this technical session one.
Distinguished guests, all the fellow researchers of this institute and the other institutes, and my dear students, a very good afternoon to all of you. It is a privilege for me to welcome you to this plenary session on a topic that sits at the very heart of modern scientific evolution, IPRs as an generating tool for biotechnology and allied fields. As we are here to discuss the intersection of law, science, and wealth creation, I find myself reflecting on a fundamental truth. Every great professional journey begins with a single spark of curiosity in the laboratory. Many years ago at KTHM College, Nasik, I had the pleasure of guiding a particularly diligent and bright postgraduate student. I remember her clearly, hunched over her lab table, working table, meticulously investigating the antimicrobial activity of synthetic quinolone compounds and nanoparticles and the anti-cancer activity. At the time, she was focused on the how and why of molecular structures.
Today, she is here to tell us how these very scientific efforts can be transformed into global assets. It is a moment of immense personal pride for me and a complete surprise for her because she didn't likely expect her former mentor to be here in this session and to introduce her in this plenary as a plenary speaker. She has evolved from that dedicated researcher in my laboratory into a powerhouse in the world of intellectual property. She is the founder and director of KB IP Services, a registered Indian patent agent. She holds a doctorate from DY Patil International University. She has a robust background in biomedical engineering and biosciences that probably comes from her husband, Mr. Swapnil, Dr. Swapnil. Huh? No? Right, right? Bioengineering background तुमचं? Okay. So she had already got expertise in this area of biomedical engineering. Her expertise now spans across plant biotechnology, food innovation, and the strategic commercialization of research. She possesses that rare blend of deep technical knowledge and strategic IP expertise. Her work has consistently highlighted how intellectual property is not merely a legal formality, but a powerful engine of economic growth and research sustainability. Beyond her consultancy, she is a dedicated academician and an influential voice for women in intellectual property, serving as a president within the Women's Indian chapter of Indian Chamber of Commerce and Industry, IP Council. Ladies and gentlemen, please join me in welcoming my former student and now a leading multifaceted expert in the field, Dr. Khushbu Bardia Burak, to bridge the gap between scientific innovation and legal protection and speak on IPR as an asset generating tool for biotechnology and allied fields. Khushbu, the floor and the laboratory is yours once again. Before we start, I would request Dr. Charan Singh Kaite sir to give a brief introduction of the speaker.
Thank you, sir. Actually, sir has already introduced ma'am, but there is a formality. Honorable Session Chairman, Dr. Petkar, sir, and dear participants, my colleague, it's my pleasure to introduce our speaker this session, Dr. Khushbu. Dr. Khushbu is the founder of KB, Indian Patent Service, and an expert and intellectual property right with a PhD in hydrogen energy sectors as the certified Indian patent agent. She has an outstanding track record of filing over 500 Indian patent applications and securing grant for more than 400 IPR. Beyond her technical expertise, she has secured over 55 lakh in funding through the SIDBI incubation program and acts as a primary mentor to more than 30 entrepreneurs. Currently, she leads the Uttarakhand Intellectual Property Council, WICCI, as its president and is an NIPAM certified IPR awareness officer. Her contributions have been recognized with an Exceptional Woman of Excellence Award 2022 and the Best IPR Consultant Award 2025 with over 40 seminars and numerous corpus publications to her credits. She begins with wealth of knowledge to this forum. We eager to it means how to write patent or how to filing patents, and it means what is exactly Indian patents, and everybody know to its modes. It means all researchers know to how to file, but not clearly how to file patents. May I request ma'am to please begin her session.
Thank you so much for such a warm introduction because it has been really, really warm to see my, mentor himself introducing me. Because how many of you are, sir, students here? Okay, all right. So you can feel me and I can feel you too. All right. Okay, so I'll be little bilingual because सरांसोबतची आठवणी माझ्या सगळ्या मराठीतल्या आहेत mostly. And whenever I used to go to sir and to show result की sir results आलेत बघायला चला. Sir used to be like very precise, मी 7.5 मिनिटात येतो तू थांब. And I used to think अजून digital watches तेव्हा आलेल्या नव्हता. How is this sir going to calculate 7.5 minutes and his biological clock used to actually take him in that 7.5 minutes and he used to come. So now I understand he had developed some nanotechnological biological clock in himself, which he might have got patented for then itself. Right? So let's all begin with the session now, and it is about the intellectual property rights. How many of you know about IPRs? Oh, I'm fine with this. How many of you know about IPRs? कोणी patent file केले आतापर्यंत? 9 faculties केले. Students? No. Do you even know about IPRs? काय आहे माहिती आहे? Intellectual property rights. The rights that you get over the property which is created through your intellect, right? So what we are going to discuss today is about the overall basis of what all IPRs are about, how and what are the most relevant to you also, and then we a little bit of when, why, and how. But before that, I want to show you this result because the previous session has been so enriching. मी बायोटेक सोडून जवळपास 8 एक वर्ष झाली आणि त्यानंतर आता हे सेशन म्हणजे ऐकल्यावर आय फेल्ट लाईक there has been so much that happened in all these 8 to 10 years, and evident enough all this is from this data also. This is the data published in 2018 actually, where India is ranking 3rd biggest research publisher, right? That means we are researching so much, isn't it? Isn't it a moment of pride? किती मोठी गोष्ट आहे की आपण वर्ल्ड मध्ये 3rd biggest researcher आहोत, because the publication number is showing that, The same year, other data was also published, is about converting the research into intellectual property rights. And can you see India now? Can you see India here? It's China, followed by US, followed by Japan, followed by Republic of Korea, European patents, and then other offices. Can you guess what should be the rank of India that year in filing intellectual properties? Basically, जे काही research आपण करत होतो, we were able to protect it through intellectual property, right? So we were researching at which level? 3rd biggest in the world. So we should have protected it at least in top 10, right? Top 20, top 30. We were at the 47th rank when it came to protecting our research. This is the story of 2018. What was wrong? If you always do what you always did, you will always get what you always got. आणि हे असेच कॉम्प्लिकेटेड रेफरन्सेस आम्हाला सर द्यायचे, ज्याच्यावर आम्ही 2-2 दिवस विचार करत बसायचो की सरांना म्हणायचं काय. And now I am also giving the same reference inspired by you, sir. Okay, so what others used to do earlier, your professors used to do same thing you are doing. Okay, because our earlier professors, earlier researchers, earlier industries did not think of protecting their inventions with the IPRs. Their newer generations also did not do that, and we had a huge gap to fill in when it came to combining our research with the intellectual property or converting our research into assets. Many efforts were taken by Indian government. 2014 Indian NIPM, National IP Awareness Mission, was launched. I was one of the IP awareness officer in this mission, and enormous sessions were conducted nationwide for creating IP awareness. And now this is the present situation. So this is the data of 2025. Again, we are ranking where? Third biggest in research publication. But in IP, again, still we are not at the same position. We are much better than before. We are in top 10 now at least. But compared to the research that we do, we are still lacking ahead. A lot gap is there. We are sixth in the world now to convert our research into patents, but still the gap is a huge gap. Why is this so? Because we are very much inspired by Neki Kar Darya Mein Dal. है ना? काम करते रहो, आगे बढ़ते रहो. हमें गीता ने सिखाया है कि फल की इच्छा मत कर, है ना? तो हम रिसर्च करते रहते हैं और आगे बढ़ते रहते हैं without protecting it. Or we also want to support our competitors. We want to support China. We want to support Korea, isn't it? And that is why we are keeping our research free of cost. Everybody come and copy our research, right? Or we have been taught since childhood की बेटा कुछ भी करना कोर्ट कचेरी मत जाना, कोर्टाची पायरी चढायला लावू नको. आणि पेटंट्स म्हणजे सगळ्यांना कोर्ट वाटतं, कोर्टाचं काम वाटतं, लीगल काम वाटतं. But trust me, it is not. It is very, very technical, as the same that you do. All right. Or have we got a lot of time and money to waste? Is it so? That we are doing a lot of research, produce something already being there in the market. If you are creating improvement, like you are increasing the shelf life of some product, right? What is the shelf life of milk? What is the shelf life of milk? Every other day we hear it in the house. दूध खराब होईल तापवून ठेव. Isn't it? So 24 hours. Ideally 24 hours. But maximum किती करू शकतो आपण store? 48 hours maximum, right? But if you can develop some technology to increase the shelf life of milk, or for that matter anything, right? Imagine how, perishable item, how much of perishable item create, or which intellectual property is appropriate for idea. So this is a basic name that I use. Think about your idea, all right? And think about, is your idea an invention? Now what do you mean by invention? Invention? New discovery. Okay. Is discovery an invention? What is discovery? Can you give any example of discovery? Discovery of pie. Okay. Okay. In case I take a soil sample from your campus and I identify a new microorganism from that sample, am I doing a discovery or invention? Okay. Okay. But I identified this new microorganism by a new process of isolation that was not used earlier. That is invention, right? So very well understood the difference between discovery and inventions. Now mind it, discoveries are not patented because they are the right of nature. But inventions can be patented. And if your idea is an invention, patent is the right protection. If your idea is about a creative expression of something, maybe in the form of write-up articles, books, poetries, pictures, diagrams, jingles, or you create some cartoons these days, caricatures are created. If you are expressing your feeling in some tangible form, written form, then copyrights are the correct protection for it. Even software codes. Codes is the conversion of your thought process into a tangible form, isn't it? So that's codes. Software codes are also copyright protected. So copyright is the right protection for it. If your idea is about creating a new design of something, autoclave मोठे मोठे खूप बोर वाटतात, है ना? आता तुम्ही काहीतरी क्रिएटिव्ह ऑटोक्लेव्ह बनवला जो दिसायला पण छान दिसतो. Now you have given an eye appealing design to it. Not necessarily दिसायला छान, but if you are creating a compact version of it, maybe you are creating a better version of it, then you are creating only the design change. The functionality remains still same, high pressure, thermal, what do you call, disinfection or sterilization. Sorry, मला बायोटिक सोडून खूप दिवस झाले आता. So sterilization. So if you create a new structure of the autoclave or any even known device, then your right protection is the industrial design protection. Okay. Then there are goodwill protecting trademarks. You know about this, right? Your logos, names of the businesses, they are protected by trademarks. IC circuits are protected by IC layouts. New varieties are protected by plant breeders' rights. Do you know about it? Being into agriculture field, if you create new plant varieties, they cannot be patented under Indian law provisions, but they are protected under plant breeders' right. Okay, and then there are trade secrets which can be protected. So first of all is patents. Patents is one of the intellectual property. It is not the only intellectual property. We are very commonly using patent. हम as a patented shirt है, हम as a patented pen है, but not everything is patentable. Okay? Only inventions, technical problem la technical solution there are inventions असतात, they are patented. All right. So if you are coming up with any technical invention, they can be patented. That gives you a negative right and an intangible property. Why am I saying it is a negative right? Negative right means you get a right to prevent others to use without your permission. Isn't it? So you are not getting anything extra, but you get a right to prevent others or using it without your permission. So that kind of right that you get to prevent others from using it is called as a negative right, and therefore it is negative right. But in reality, when you experience IPRs, it's not negative at all. Patentability needs to satisfy these three criteria. So think about your invention, whatever maybe your MSc projects or anything that you are doing. Think about, is it novel? Is it non-obvious? And is it industrially applicable? So the third criteria is simple. Is it useful? Isn't it? So we just need two criteria. Is it novel and is it non-obvious? Novel, what is novel? New, something that was not existing before. It can be anything. It can be a product, it can be a technique, it can be a process, it can be a combination that was not used before is novel. But now what is non-obvious? That process combination should not be just the obvious replacement of already available. Like what do you use for surface sterilization? 70% ethanol. If I come up, Eureka, I have found a new surface disinfectant. I'm using 70% propanol. Am I doing invention? You will say, obviously, yaar, ethanol use karu shakto, to propanol use karu shakto. Isn't it? Because these are obvious replacements of each other. Right? Efficacy, I don't know if, sir, is comparable to both of them, but they are comparably obvious replacement of each other, isn't it? So if it is an obvious replacement of something already known to the person skilled in the art, then it is not patentable. But if it is not obvious replacement of something already available, so till date you had been using alcohols for surface sterilization. Now you are coming up with ethers maybe, or some other compounds that were never used before as surface sterilant, then yes, it will be novel and non-obvious. Okay. By now you might have already, okay, and there is also one criteria which has never been documented anywhere, that it should be disclosed in an application in a prescribed manner. The Indian Patent Act 1970 मध्ये जे काही rules and regulations दिले आहेत, त्या फॉर्म मध्येच पेटंट लिहिला गेला असेल तरच तो पेटंट होतो. So we have experienced many inventions which were novel, useful, non-obvious, but they never got a grant of patent because they were not disclosed in a proper manner. All right. So this is also a very important term to remember, that they should be disclosed very properly. Now, by now you have understood novelty. I just wanted to give you one example of how to evaluate non-obviousness so that you can evaluate the patentability of your invention on your own. So consider your invention as a claimed invention. Okay? आता प्रत्येकाने आपल्या आपल्या डोक्यात आपला रिसर्च घेऊया. Okay? And I know very well that all the professors, I know Maya ma'am also asks you to do a detailed research before actually starting the research. That's called review of literature, right? During which you find a lot of articles which are exactly similar to your research, isn't it? So consider that as the closest prior art and your invention as the claimed art. Now what you do is compare and identify if there is some difference. Are there some differences in the chemicals that are used? Are there some differences in the processes that you are going to use which they have not used? All right? Find out what are the differences. If you find out that there is no difference, it is exactly same compound, same mixtures, same process, same product, same efficacy of the product also, then your invention lacks novelty. But if there are differences, then yes, your invention has novelty. Now let us check if it is non-obvious. If that replacement of differences, that differences that are present in your claimed invention and the invention that is closest prior art of your, if that difference is obvious to the person skilled in that art, and you are the skilled person in your art, isn't it? You know the best about your art. And if you feel like this is an obvious replacement what is been already done, then it lacks it lacks inventive step, which is called as obviousness. And if that difference is not a very obvious replacement, but a new thing, okay, the difference is there, and that difference is not obvious difference. It's a new difference. It's an unobvious difference. Then your invention has both inventive step as well as novelty, which means you can go ahead for patenting your inventions. There is this threshold rule for patentability, but there is a big list of what cannot be patented. So this is under Indian Patent Act, that frivolous and something that claims very obvious and contrary to the well-established natural laws cannot be patented. So if you invent a perpetual time machine, okay, sorry, it will not be patented because it is against natural law. All right, so anything against natural law cannot be patented. If something is contrary to the morality or injurious to public health, cannot be patented. So if you identify a new virulent of, virulent or any virulent, right? If you identify a new virulent and if you feel like patenting it, not possible. Why? Injurious to public health and immoral to get it patented, isn't it? If you happen to invent a new gambling device, maybe a new, hacking software, hacking codes, then these, although it is ethical hacking or unethical hacking, in both ways it will not be patented because they are considered as immoral to be patented. Anything which is just a discovery or a scientific principle discovery, so you happen to invent Newton's 6th law, 7th law, 8th law, calling it Pattil's 3rd law, Pattil's 5th law, may not be patented because they are just discoveries. Any mixture of already known things which is not giving us any unobvious result. So you just mix A with B and C and all the properties of A, B and C are mixed, nothing additional to it. Okay, it will not be patented. But if you are mixing A, B and C and something additional in addition to the properties of A, B, C is coming up, okay, then yes, that can be patented. So even if I'm saying these are the non-patentable list, identify the differences between the words, read between the lines that yes, something can be patented if you create a new, if your combination is giving new efficacy, improved efficacy, or new benefit that the combination usually doesn't give, okay, then it can be patented. Then the mere rearrangements. So one fine day you open your microscope, and you just reassemble and create a new microscope saying you have created it. That's not patentable because it is just mere rearrangement unless it is giving some more advantageous result than the earlier. Method of agriculture and horticulture. Now, I just want to pause here a little. There is a big buzz that agriculture and horticulture related inventions are not patentable. But read the sentence very carefully. It is method. Method of sowing, plucking, winnowing, harvesting cannot be patented. Because of course it's immoral if you get the monopoly rights on sowing. Nobody will be able to sow the seed only. Isn't it? But if you are creating a new tool for sowing, a new method for winnowing, a new technology for harvesting, yes, they are patentable. Only the method of doing agriculture and doing horticulture is not patentable. All right? Related to atomic energy, for a very obvious reason, they are not patentable. Inventions related to, again, I would like to bring your attention to this sentence. Any process for medical, surgical, prophylactic, diagnostic treatments of humans or animals cannot be patented. What is not patentable? Process of diagnosis. If you create new tools of diagnosis, yes, they can be patented. If you create new sensors for diagnosis, yes, they can be patented. If you create new genes as the sensors, yes, they can be patented. It is only the method. कुणीतरी डॉक्टरने ओपन हार्ट सर्जरीज पेटेंट करून घेतली तर बाकीचे डॉक्टर्स काय करतील? Right? Surgery's method of treating the patient cannot be patented. But technologies, tools, and even small suturing machines and devices that are used in medical field, yes, they can be patented. For biological invention specifically, you need to deposit if that invention, which is including some biological material, is not a publicly known thing. Then you need to deposit that into National Biodiversity Authority Board. And if it is already a publicly known thing, then you can just get the approval of using it. Like if you are using the coconut for doing some kind of research and now you want to patent that research. Now because coconut is a well-known thing, you can just take the approval of the Biodiversity Board and go ahead for patent filing. If you are you have developed a new microbial breed and you are sure that it's not a well-known type of microorganism, and you will need to deposit it into the National Biodiversity Board before. And there are many, in Pune also there is MCC, there is MTCC in Chandigarh, and there is NAIMCC in Uttar Pradesh also. Okay, we discussed, we heard from the previous speaker that a lot of collaborative research is going on. And why IPRs are very, very important when it comes to collaborative research. I would like to take the attention of all the research faculties here who are engaged into collaborative research, that a lot of policies of Indian IPRs are not similar with the other country IPRs. The difference being intellectual property rights are governed by acts, and acts are governed by laws, and laws are different from country to country, right? So IPRs are territorial rights, which means if you get a patent in India, nobody without your permission will be able to use it in India, but anybody can use it in US. And the vice versa is also true. If you think like हम international patent लेंगे, बरेच लोक माझ्याकडे येतात मॅम ते इंडियन पेटंट वगैरे खूप कॉमन आहे, मला आता US पेटंट हवंय. What will happen if you take a US patent but your client base is Indian? US में कोई कॉपी नहीं कर पाएगा, but India anybody can freely use it. Okay, because intellectual property rights are territorial rights. Our patent system is very much influenced by European patent system. Reason being we are inheriting all the laws from Britishers. But there are many differences also. Like in case of European research, European patent, a researcher has 1 year from the time of public disclosure to file a patent. Which means if you accidentally publish your research first and then you think of, I should have got it patented, European and Indian system still gives you 1 year to get it patented. आय होप दिस इज क्लिअर. तुम्ही रिसर्च पेपर पब्लिश करून दिला आणि नंतर रिअलाइज झालं की मला हे पेटंट करायचं होतं. जनरली तसं करू नये. यू शुड फर्स्ट पेटंट अँड देन पब्लिश. ओके, बट बाय चान्स इफ यू हॅव पब्लिश्ड इट बिफोर, Indian and European systems still give you 1 year duration to file a intellectual property patent for it, but that is not true for US. In case of US, plants are patentable, but in case of Indian and European systems, plants are not patentable. In case of US, first person who invents has the right to get it patented. In India and Europe, first who files the application has the right to get it patented. So if I and you are researching on the same project, and if I file it before you do, and you keep on waiting for result येऊ दे, छान result येऊ दे, अजून trials घेऊ दे, then you are losing it. Okay, so first to file is patent holder in case of India and Europe. Can I have a global patent? No. Do we have global laws? UN सोडून बाकी कुठले laws आपले global laws आहेत? No. And if something is governed by law, can we have it globally same? No. I just gave some of the examples. Like plants are not patentable in India and Europe, but they are patentable in US. So because laws are different from country to country, you cannot have one patent which gives you protection all over the globe. If you think like you want to protect your invention in India, Australia, and Europe, and maybe China, you will have to file individual application in all these countries. Or there is a provision that you file a PCT application. PCT is actually a treaty which is signed by 200 plus countries, India being one of them, which agrees that एका तारखेला फाईल केलेलं ऍप्लिकेशन त्याच्या पीसीटीच्या अंडर जितके कंट्रीज आहेत सगळ्यांना त्याच तारखेला फाईल केलं गेलं असं कन्सिडर होईल. I hope you got a little bit of it. If you file a PCT application for an invention on 1st of January, okay, then you go to China, travel to China, 2-3 दिवस आराम केला, जेट लॅग घालवला आणि एक आठवड्यानंतर 8th of January ला you file a Chinese patent. Now your priority date changes. But if you have filed it through PCT route, your priority date remains 1st of January in all the countries that you want to file under PCT. Okay, so it is just a provision to save the priority in all the countries because you get a duration of 30 months to go and individually file in those countries, and the date of priority remains same. So if anybody files after you, will not have a priority, but you will have a priority. But you cannot have one single patent which give you global protection. That's not possible. Okay. Process of filing, if you have filed already a patent, it starts with filing a provisional or a complete specification. If you are completely ready with your invention with results and everything prepared, you can file a complete specification. If not, you are ready with a blueprint and yet to, fully create a shape out of it that you feel like it's yet some time is required to actually get some results, then you can file a provisional specification. Provisional specification gives you a 12 months duration to replace it with the complete specification. All right. So if you file it with the complete specification itself, it is published after 18 months automatically. 1.5 वर्षानंतर पब्लिश होतं. प्रोव्हिजन आहे you can get it early published. 1 महिन्याच्या आत पब्लिश करून घ्यायची प्रोव्हिजन आहे. You can file for request for early publication and get it published early. This publication has nothing to do with your research publication. Being into academics, I have seen many people showing off patent published in their CV, which has zero importance. Even if I write twinkle twinkle little star and I submit it, it will be published. Because this publication is happening without any examination. Examination is going to follow this process. After the publication, you file a request for examination and then it will be examined. If you don't file a request for examination, it will not be examined. So this publication that happens automatically, which I said, or can be done earlier, is only to have, any pre-grant opposition. If anybody wants to oppose you that, you cannot patent it because I have already published this data. सो दॅट काइंड ऑफ ऑब्जेक्शनची विंडो देण्यासाठी प्री ग्रांट पब्लिकेशन केले जातात की वी हॅव रिसिव्ड दिस ॲप्लिकेशन बाय दिस इन्व्हेंटर अँड दिस इन्व्हेन्शन सेज अ लिटिल बिट एक ॲब्स्ट्रॅक्ट फक्त पब्लिश केलं जातं सो ॲज टू कॉल इफ एनीबडी हॅज एनी ऑब्जेक्शन्स राईट बट it doesn't mean anything from your asset point of view. Once you file a request for examination, examination happens, you receive a FER. This has nothing to do with FIR. People come up frightened, Madam, FIR ali. FIR nahi hai. This is first examination report, FER. You get a duration to answer the FER, which means तुम्ही research paper कधी केलाय का? Communicate. Students, I'm not going to ask faculties because I know they have tons. Research students, right? So कधी review येतो reviewers चा की डायरेक्ट पब्लिश होतो पेपर रिव्ह्यू येतो ना सो तसंच सेम फॉर्म मध्ये रिव्ह्यूवर काय मागतो की वाय डिड यू इन्क्लूड दिस सेक्शन यू शुड हॅव डन दिस यू शुड हॅव ॲडेड दिस explain me why this is so, right? Same this kind of explanation comes from intellectual property office. They either want some explanation to what we have probably fallen short of, or they come up with some already available prior art which they feel like are similar to our invention and therefore our invention cannot be patented. So in those cases, we get a duration of 6 months to respond to those. Even if we we are not able to respond in 6 months, we get 3 months additional. And within 9 months, you can furnish all the requirements that are being quoted in the FER. If you don't, your application is considered abandoned, which means you are no more interested in getting your patent. If you do, your patent is granted. Now, this grant means your patent is granted, and this grant gives you the duration of 20 years of protection. All right? This 20 years of protection start from this date. This doesn't start from this day. All right. Examples. So when I was into academics, this girl came to me for evaluation of some other project, but she brought some sweet for me and asked me, "Ma'am, can you guess the ingredients I've used?" And I could not, and none of our panel members were able to. And when she disclosed, we were in surprise that how can this be made by using this kind of ingredient? And I just told her, just keep it here and don't disclose it anywhere and we will get it patented. And not only her, but also my co-panelists were surprised that food can be patented. What are you saying? And then I took it as a challenge, right? And then I got it patented and she holds a patent. And many of the corporates in Hinjivadi nowadays, जे न्यू इयरला फरेरो रॉशर वाटायचे ते आता तिचं स्वीट वाटतात. We have got a patent for the process also. A very small change in the process. Hydrogen energy वर प्रचंड काम चालू आहे, isn't it? I did also my PhD in the production of biohydrogen. But there is a huge problem in storage of the hydrogen produced. Produce तर खूप करता येतो आम्हाला, पण स्टोर नाही करता येते, isn't it? And स्टोर जरी करायचं म्हटलं तर नाही, तो गॅस प्रोड्यूस करायचं नाही म्हणते मी, ज्याच्यावरून तुम्हाला हसायला आलं. हायड्रोजन गॅस. अँड this method was only to increase the adsorption capacity of the hydrogen. Kitti small invention आहे, just increase in the adsorption capacity. All right, but it will create a huge difference if it is converted into technology in the industries, right? And therefore that invention also we got patented. This person came to me as a healthcare facilitator who is also into research. Who had created a software by machine learning. Now, what is machine learning? You make the machine learn something. माणसासारखं काहीतरी त्याला शिकवायचं आणि मग त्याच्याकडून रिझल्ट एक्सपेक्ट करायचे, isn't it? So, she had created Alzheimer detection tool using multi-modality neuroimaging biomarkers. So, she trained her machine with different images of Alzheimer disease patient scans, okay, and wanted to get it patented. Now, generally, this kind of inventions cannot be patented because they come under the category of 3K, which says that only the softwares per se cannot be patented. All right, and this was something involving softwares, but we got it patented because there was a technical problem and the technical solution, right? And if a technical problem is coming up with a technical solution, even software related inventions can be patented. So this was coming up with improved biomarker extraction accuracy, improved feature alignment, improved diagnostic sensitivity, improved resolution, improved feature fusion. So because of all these improvements, this technology got also patented. Then this is the second intellectual property rights, which is called as industrial design. First, patent, done. This is second, industrial design. Anything and everything lying around you, with you, is a product or a property of industrial design. Everybody has a phone, huh? Without looking at the logo, just phone's shape पाहून कळतं ना कुठल्या कंपनीचा आहे? Because those companies have got their designs protected. Okay, anything right from your jewelries, chappals, your utilities, bags, your outfits, all are industrial design protected by the manufacturers. This is a very good example that I always like. Have you ever bought any nasal inhaler other than this? Because I have not. Have you ever bought? No. Why this company could, completely vanish out all the competitors from the market? Only one design change. What? Hearing ऍड केली फक्त इनहेलर मध्ये. And only this small design change vanished out all the competitors from the market, that till date if I ask for inhaler, only Vicks comes up. Isn't it? So design is that important. If you are having equal price and equal technically similar product in the market, you have to buy, and you go and buy only something which is designing, who's designing appealing. Isn't it? मार्केट मध्ये गेल्यावर दोन तेलाच्या बॉटल जरी दिसल्या, सेम क्वांटिटी, सेम प्राइस, सेम क्वालिटी, आपण जी बॉटल चांगली दिसते ती घेतो. ऑल्दो आपण बॉटल नसतो खाणार, तेल खाणार असतो, राईट? बट वी आर सो इन्फ्लुएन्स्ड बाय द डिझाईन्स that yes, we buy because of the designs. And if design is so important, it should be protected. The biscuits that you have in the morning, marigold biscuit, त्याच्यावरची जी डिझाईन आहे ना, that is protected. KitKatचे जे वेफर आहेत, that design of that wafer is protected. Coca-Colaची bottle, even if I erase out the label of it, you can still identify. For that matter, any bottle, if I remove the wrappers of Bisleri, Kenley and Himalaya, you can still by the design of the bottle identify which company bottle is it. Isn't it? That's because design lays a huge impact on the customer's minds. And if you are into designing something, you should protect it, because it is going to create a huge difference. These are some examples we got registered for our clients. So this is a soil sensing device which was created. This device was also manufactured by us. He came up to us with the technology only, and we got it manufactured. The pesticide spraying drones. Now this technology is very commonly used these days, right? So technology is nothing new, but the design of the drone was new. Okay, so he did not patent the technology, but he got the design of the drone registered. Understood the difference, right? Then the whole system, solar energy storage system, the whole system design was also protected by one of a university which is very greatly engaged into solar kind of research. This is a very recent grant for design we have got for bioreactors for microalgae growth. So reactors चे design सुद्धा design protect होतात. This is a UK design patent. So a client of ours was going to supply his materials into United Kingdom, and therefore he wanted UK patent, UK design registration, sorry. So we got it patented. It is a device for sewing. There is a nematode catching fluid which has created, and that needs to be sewed. Okay, so that sewing device, we got it design protected, industrial design protected in UK. So this is a modular vertical garden pot. सोप्या शब्दात हायड्रोपॉनिक जे pots असतात, that are protected by industrial designs. The third intellectual property right, maybe very commonly you might have heard, GI, geographical indication. Mahabeshwarchi strawberry, Kolhapuri chappal, सोलापूरची चादर, राईट? अजून काय काय भरपूर. इफ एनिथिंग ओरिजिनेट्स फ्रॉम सम specific geographical location can be protected. Uttaranchal tejpan, Kashmiri saffron, Kumaon churia oil, Mysore petal leaf. These are one of many GIs that are protected in India. Trademarks. कुछ मीठा हो जाए? Which company? Okay. जिंदगी के साथ भी, जिंदगी के बाद भी? LIC. Okay. The Taste of India. And what are these two? Last? कुठे लिहिलंय? कुठे लिहिलंय? इथे लिहिलंय की हा लोगो दिसला म्हणजे स्टारबक्स आहे. हा जर लोगो हा आपल्याला कुठे रेगिस्तानमध्ये पण दिसला ना, इतकं सुकून वाटतं, इतकं शांत वाटतं. हा सगळं कळतं, कसं आहे, काय आहे, किती ऑप्शन्स मिळतील, फर्निचर कसं असेल, इतकं सुद्धा माहिती. Isn't it? Because the logo creates a huge impression of your business on the client's mind. All right? Not only logos. आता मी एक जिंगल ऐकवते, मला सांगा कुठल्या कंपनीची आहे. टिंग टिंग टिडिंग. इतके अभ्यास करतात तुम्ही सगळे? रिपीट. टिंग टिंग टिडिंग. ब्रिटानिया. खूप अभ्यासी मुलं आहेत. So even jingles are protected. Nokiaची ringtone was protected. Jingle protected. Even तुम्हाला माहिती आहे, smells can be protected. Recently, Indian Trademark Office gave trademark for smell. There is a tire company which manufactures a tire which smells like rose. Tire जेव्हा खूप जास्त गरम होऊन जातात ओव्हर ड्रायव्हिंगमुळे, तर त्यांचा एक स्मेल येतो. तो अवॉइड करण्यासाठी ते इन्क्लुडेड सम कंपाऊंड्स ज्याचं त्यांनी पेटंट घेतलं, पण त्या स्मेलचं सुद्धा ट्रेडमार्क आहे. ही एक फेमस केस मला नेहमी सांगावीशी वाटते. चितळे बंधू सगळ्यांनी बाकरवडी खाल्ली आहे. डू यू नो दिस वॉज नॉट प्रोटेक्टेड बाय ट्रेडमार्क. आणि आता खरी स्टोरी सुरू होते. गेल्या वर्षी मार्केटमध्ये आले हे दुसरे प्रॉडक्ट, चितळेच बाकरवडी. ओके. आणि चितळे बाकरवडीचा, ओरिजिनल चितळे बाकरवडीचा सेल खटकन ड्रॉप झाला. का? देयर वॉज अ ह्यूज डिफरन्स इन द टेस्ट. बट पीपल लाईक अस डिड नॉट अंडरस्टँड की ह्या दोन डिफरंट कंपनीज आहेत. वी जस्ट स्टॉप बाईंग चितळे बाकरवडी होऊन, कारण टेस्ट सारखी येत नाही. अँड देन चितळे बंधू रिसर्च की असं का झालं, अँड देन दे गॉट टू नो की देर इज समबडी नोन एज चितळे स्वीट होम हु इज सेलिंग द चितळे बाकरवडी. बट दे कुड नॉट ऑब्जेक्ट बिकॉज त्यांनी स्वतःच ट्रेडमार्क घेतलेलं नव्हतं. नाव दे हॅव फाइल्ड अ ट्रेडमार्क फॉर इट. ओके. So it is always better you get it protected. The last question, people ask us, why do you charge so much? Now tell me what is claimed here. When we write patents, we convert it into techno-legal language. Can you guess what is written here? इथे स्क्रीनवर दिसत असेल तर तिथे बघू नका, इकडेच बघा. Should I read it? Okay. 3 Idiot वाला scene आठवेल. A spherical elliptical fried crisp hollow bread that is made by combining a mixture of one or more flours selected from a group of consisting of chickpea flour, wheat flour, rice flour, corn flour, semolina with one or more spices or water, which at the time of serving would be taken up as a spherical hollow bread with a hole made at the center, filled up with a mixture of one or more ingredients selected from a group of consisting of potatoes, onions, chickpeas, lentils, bean, along with one or more spices and liquid additives contained within a small hole in the fried dough ball, wherein the liquid additive is selected from a group of consisting of tamarind chutney, mint chutney, coriander chutney, yogurt and lemon juice as per consumers' preferences. So when you give us an invention to protect the pani puri, we convert it into this much. Okay, that is why the question is, and this story I just told, this is the 11th standard student who came to us with this invention. छोटासा एक ब्रेड बॉक्स आणि ते आर्डिनो वगैरे लावून ते घेऊन आलेला होता तो. And खूप क्रूड फॉर्ममध्ये होतं हे इन्व्हेन्शन, बट नॉवेल होतं. वी फाइल्ड अ पेटंट फॉर इट. देन वी क्रिएटेड अ कॉम्पॅक्ट व्हर्जन ऑफ इट. ओके, दिस वन. वी प्रोटेक्टेड द इंडस्ट्रियल डिझाईन राईट्स फॉर इट. सॉफ्टवेअर जो होता याचा अॅनालॉग टू डिजिटल, त्याला कॉपीराईट घेतलं, त्याच्या कंपनीचं नाव ट्रेडमार्क केलं, अँड नाव दिस 11th स्टँडर्ड स्टुडंट इज सेलिंग इट ऑन द डिजिटल मार्केट platforms. So if a 11th standard student can do, you can also. Thank you. If you have any questions, I would be happy to answer. Yes. So it was just the, yes. So it was just for the sake of example that we have written it. Although Panipuri composition cannot be patented now because it's already available prior and publicly available prior. Any other questions? कळलं की काहीच कळलं नाही? मग क्वेश्चन विचारा, नाही तर मी उतरणारच नाही इथून, लंचलाच नाही जाता येणार. येस. मॉडेल्स, व्हेकल्स. स्कूटी पेप वापरली का कधी? नाही वापरली आहे. स्कूटी पेप जेव्हा मार्केटमध्ये लॉन्च केली, ती सिस्टम स्कूटी पेप जेव्हा मार्केटमध्ये लॉन्च केली गेली, त्याच्या आसपासच्या म्हणजे त्याच्या आधीचे नंतरचे जे काही त्यांनी डिझाईन व्हेरियंट्स क्रिएट केले असतील ना, 27 डिझाईन्स पेटंट केलेल्या होत्या. म्हणून त्याच्यासारखी दिसणारी सुद्धा कुठलीच मार्केटमध्ये स्कूटर आली नव्हती. त्याच्यामुळे त्यांनी खूप वर्षापर्यंत त्यांचा दबदबा मार्केटमध्ये ठेवला होता. So answer to your question is, every automobile company cannot survive without intellectual property rights. त्यांच्या टेक्नॉलॉजीज पेटंट होतात, त्यांच्या डिझाईन्स प्रोटेक्ट होतात, त्यांची सॉफ्टवेअर्स जे इंटिग्रेटेड असतात ते कॉपीराईट होतात, त्यांचे छोटे इव्हन डिझाईन, बल्बची डिझाईन, आता ती मर्सिडीजचे बल्ब असे साईडला लावणे, राईट, आय काइंड ऑफ जॅगवारचे, सॉरी. So bulk designs, every small ingredient which they are going to change in their whole process is patented. Yes. There are two different IPs related. When the model structure is similar, design rights are applicable. Yes. Features can be same if they are structurally looking different, if their eye appealing is different. Irrespective of their features being the same, they can own different design rights.
Okay, ma'am. Last question from my side. What is the exactly design patent and utility patent?
So design patent and utility patent ही इंडियन कन्सेप्ट नाहीये. इट इज US कॉन्सेप्ट. तिथे पेटंट हा एकच वर्ड यूज करतात सगळ्या आयपीजला. बट इन इंडिया पेटंट्स आर गिवन टू टेक्नॉलॉजीज. डिझाईन्स आर प्रोटेक्टिंग द डिझाईन न्यूनेस, म्हणजे whatever is applicable, whichever is judgeable by purely by visualizing, that is protected under designs. Usually इंडियामध्ये त्याला डिझाईन पेटंट म्हणतात, बट इट इज इंडस्ट्रियल डिझाईन, नॉट डिझाईन पेटंट. That's the not right terminology to be used in India, but meaning is same.
पेटेंट फाईल करायचं असतं ते एक्झॅक्ट फीस किती लागते? हाऊ मच फीस पे फॉर पेटेंट फाईल?
पेटंट फाईल करण्यासाठी खूप फॅसिलिटीज इंडियाने दिलेल्या आहेत फॉर गव्हर्नमेंट इन्स्टिट्यूट्स लाईक यू. फर्स्टली, यू आर एलिजिबल टू गेट इट अर्ली पब्लिश्ड, अर्ली examined also, not only early published but early examined also, which is called as expedited examination route. Okay, so filing ची फीज असते 1600 रुपये for government any academic institute, 1600. If you file for early publication, 2500. If you file for a regular examination, 4000. If you file for expedited examination, 8000. That's it.
And consultant fees?
Depends from project to project, because we convert it into technical, from technical to techno-legal. And then there are processes also which we call prosecution. So the legal part is the prosecution. The patent agents like us handle on your behalf. So that prosecution fees is charged based on case to case, because not every case will face opposition. Not every case will go into hearing. So it depends from case to case.
Okay, thank you very much, ma'am.
Yes, thank you so much, sir.
Uh, Ma'am has covered all these things related to IPR. May I request Peter sir, please give valuable feedback for this session.
Thank you, Dr. Khushbu, for that very insightful and strategically grounded presentation. Watching you speak today, I am reminded of our time at KTHM College. Back then, you were looking at the antimicrobial activity of quinolones using an agar plate and microscope. Uh, Today, you have shown us how to look at biotechnology through the much wider lens, the lens of economic valuation and global protection. As a chairman, I'm struck by a few key takeaways from your presentation that I believe we should all carry back to our institutions and our laboratories. So I'd like to highlight three critical points from her talk. Number one is the bridge from bench to the bank. Dr. Bhurat has shown us that a patent is not the end of a research project, but it is the beginning of its life as a commercial asset. Number two is the versatility of biotech IP. Whether it is biohydrogen or whether it is food technology, the legal framework is what gives the scientist the freedom to operate. And the third important point is the importance of strategy. In her words, innovation without protection is merely a gift to your competitors. On a personal note, there is no greater reward for a teacher like me than to see their students surpass them in expertise and lead all the way for the next generation. Khushbu, you have navigated the journey from biotechnology and chemistry labs in Nasik to the forefront of IP consultancy with grace and brilliance. You have truly demonstrated that while science creates knowledge, IPR creates value. Firstly, you have successfully shifted the narrative from IPR from being a defensive shield to an active asset in biotechnology, where the R&D cycles are long and investment heavy. Your explanation of how patents and trade secrets can be leveraged for licensing, valuation, and attracting venture capital is particularly timely. Secondly, I also appreciate your emphasis on the allied fields, whether it is food technology, agriculture, or environmental biotech. You demonstrated that the commercial value of an idea is only as strong as its legal boundary. Your insights into biohydrogen production, the sweet protein bonds, the traditional knowledge protection served as an excellent case studies on how nuanced IP strategies or complex IP strategies can protect even the most complex biological innovations. Finally, your message was very clear. For biotechnology to truly serve the society, it must be sustainable, and there is no sustainability without the protection of the intellectual labor that drives it. You have given us the roadmap on how to move from publication to patent and eventually to the product. So on behalf of the organizing committee and on behalf of the audience, I thank you very much for your time, your expertise, and for sharing your vision with us today. Let's give Dr. Khushbu Bhurat one final resounding round of applause. Thank you very much.
With this, I take the permission from the chair that the technical session one has been concluded. Now there is this lunch break. Everyone, please go and have your lunch, which is arranged at the library of Government Institute of Forensic Science. And all have to gather to the respective designation wherever your presentations are aligned. Okay, so just to make it a point, see for offline oral presentation research scholars, the auditorium is the venue. For faculty and UG category, biophysics department. For PG oral presentation, Department of Microbiology and Department of Biotechnology. And for the poster presentation, auditorium, aisle is been arranged for your posters. Okay, so according to the code that has been given, you can display your posters. And everyone, please again make it a point to come back here at 2 o'clock.
कहाँ है यहाँ पॉज ऑप्शन अभी जैसे आपने स्टॉप करा स्टॉप नहीं करा था स्टॉप नहीं होता पॉज सेशन पॉज नहीं होता कभी एंड करना पड़ेगा उसको कैमरा को स्विच करके वो जो आपने टेम्पलेट दिया वो टेम्पलेट आ जाएगा चलेगा ठीक ये चलते रहेगा ना म्यूजिक हाँ म्यूजिक चालूच राहील ना चालूच ठेवतात लाईव्हसाठी हाँ सिर्फ उस पे बैनर को फोकस करके लगा दो सिर्फ बैकग्राउंड नहीं दिखेगा हाँ वैसा करो सिर्फ बैनर पे फोकस कर अभी उधर गुलाबी चालू नहीं करा है अभी उस साइड से प्रेजेंट उधर बैठेंगे लैपटॉप अगर आपका इधर पे भी लगा सकते हैं तो पेन ड्राइव भी लगा सकते क्या बोल रहा कॉल करना है क्यों
क्या लाइट अभी चालू रखी बाद में बंद कर लाइट वो काइटे सर ने आगे फंस गया था तो उससे स्ट्रेट हो गया वो प्रोजेक्टर की स्क्रीन यूं है ना तिरछा इसीलिए तिरछा भी बनती है मैं बोली तो थी मैं वो ये है आगे खसका दिया था उसने तो स्ट्रेट हो गया अरे वो जो है ना उसको ऐसा था वो ऐसा करा अच्छा बैक को हाँ मैं कोई तो बोल रही थी उसको सीधा देना कब करा तो ले नहीं गए गाड़ी से लैपटॉप लैपटॉप लाए थे खतरनाक तरीके से क्यों चार्जर तो डिक्की में डाल दिया लैपटॉप में डिक्की में डाल दिया तो आया नहीं तू आगे फुटेज में रखा है बैग ले जाता ध्यान से उतर गया था मैं तो ध्यान में था हाथ में पकड़ के दे मोबाइल चोरी उसमें रख लेता ना डिक्की के उसमें अरे सुनना गाड़ी की चाबी ले ली से हाँ अरे मैंने देखा मैंने 210 के पैसे बचाए नाले मैं भूल गया कल वो ट्रांजैक्शन चेक करे ना मैंने तो उसमें देखा तो मानने को तैयार नहीं कर मैंने ही कम डरवाया बेचारे को इतना सुना दिया किसने सुनाया मन ने नहीं लेके तो गया है वो भी ऐसा अच्छा नहीं है उसने खैर पर इतनी नहीं कि पूरा पेट खाली हो और वो खत्म होना बनता था ठीक है क्या अमाउंट तेरे पास है कॉलेज है क्या नहीं रे मेरे पास ही रहता है मेरे पास कहीं नहीं जाएगा सेफ है मेरे पास तेरे से ज़्यादा सेफ रहेगा मेरे पास से अभी हिसाब लगा हमने कुछ प्रॉफिट नहीं खर्चे निकालने ना अभी अपने और भी तो करे थे अभी बीच में बैनर में से 200 रुपये बने तो 200 रुपये कहीं ना कहीं खर्चे में भी निकले उसमें उसमें से कुछ रुपए अभी अपने पास टोटल अमाउंट 10,000 रुपए आए थे, तो 1000 में से 20 रुपए का तो हो गया। उसके अलावा 500 को मेरे पास कैश में पड़े हैं, 500 जो है ऑनलाइन उनका 1000 रुपए है मेरे पास।
अब अपने पास 1000 रुपए है, तो कोशिश करो कि जब तक कि इंदौर जा रही हो अपने को और कुछ मांगना नहीं पड़े। प्रज्ञा पूनम का प्रेजेंटेशन होने वाला है, वही आ रही क्या, मैंने बोला जादू
कहाँ पे है
बायोलॉजी वाली
मैं देखता दोनों साइड करता हूँ देने जैसा बात, क्योंकि वहाँ पे एक बार स्लाइड लगा के दे देगा, ताकि उसके बाद पॉइंट नहीं कर देता सर।
Hello. Shortly, we are going to start the oral presentation. So those who are going to present should submit their PPTs to the slide presenter. So put the code, whatever code has been assigned to you, on the file and then submit.
Hello, kind attention, please, everyone. Uh, those who are going to present the oral presentation, uh, the first candidate, that is OFO1, I am sure that he or she has submitted the file. So we will start with the session. So those who are remaining to submit, just take your seat. One by one we will call and then we can submit it. Hello. Oh. Thanks. So good afternoon, one and all. We are going to start the session. Before we start the session, I would like to welcome few individuals who are key to this session. We have with us Dr. Nitin Ardapure, who is head of Department Biotechnology, Microbiology at Swami Vivekanand College, Aurangabad, or Chhatrapati Sambhaji Nagar. So I would like to invite him on the stage and would like to request Suchita madam to just welcome him. What? So I want you all to give a big round of applause, please. Thank you, ma'am. Once more, ma'am, we would like to also welcome our second judge for the session, Dr. A.N. Salve, sir, who is professor at Botany Department at Institute of Science, Chhatrapati Sambhaji Nagar. So I would request once again to Sujita madam to welcome him. Thank you, ma'am. So I would request the first candidate, OFO1, whoever is coded OFO1 to come onto the stage. I'd request the person who is operating the slide to initiate the PowerPoint presentation. Ali, better, right? Yeah, ma'am.
मेरा फोन तेरे पास है ना
Yeah. Uh, the total time given for presentation is 5 minutes plus 2 additional minutes. So you have to wind up in 7 minutes. So that would be the timeline that would be given for all. So kindly follow it.
Hello. 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 Good afternoon, all. My topic is cervix shield, integrating HRHPV genotyping with the structure-based drug discovery for the cervical cancer prevention and targeted therapy. Background and why is it so? Cervical cancer is the fourth most common cancer in the world, and it is the second most common cancer in India, contributing to 25% of the global mortality. One woman dies every 8 minutes of cervical cancer as per the ICMR statistical data. And as per the India Today Network, we continues to lose every 1 hour 9 women. As per Hindu Express, only 1% of the women are screened for the cervical cancer, and only 1.3% of the women are vaccinated in India, as per National Family Health Survey. So why is it so? Because the HPV infection remains undetected for the years. Treatment starts after the cancer develops. As there is no targeted screening and treatment, current emphasis is only on the pap smear and the 1618. So the though the American database shows not two genotypes, but still the clinicians are focusing on only on the 16 and 18. Screening is symptom based. When the cancer comes to the third stage, then the treatment starts. Till then, only the pap smear analysis is done. Low in-house production of the kits are available in India, and lack of awareness and affordability barriers. Today I came up with I have designed a kit with the screen and treat model with the objective of the molecular screening that early detection of the extended 14 high-risk genotypes using the RT-PCR which aligns with the WHO 90-70-90 model by the year 2030, which says that 90% of the women should be vaccinated, 70% of the women should be screened for the cervical cancer, and 90% of the women should be treated for the cervical cancer. Now I have done the comparative assessment of the accuracy, turnaround time, and clinical impact of the pap smear versus RT-PCR, so as to see why the conventional methods are still being used in the diagnosis.
Now I have done molecular treatment, which is a treatment model for my project. Identified the most prevalent HRHPV genotypes from the screening data. Performed the molecular docking plus ADME analysis. ADME analysis means the absorption, digestion, metabolism, and excretion for the particular docking to identify the lead molecules for the targeted HPV genotypes, bridging the screening and the targeted identification. That is the genotype-based drug discovery. Following this methodology from primer designing, DNA extraction, quantification of the DNA selection of the detectors, analysis and interpretation. This study was conducted from October 2022 to June 2024. This was the inclusion criteria and exclusion criteria. The women with the symptoms like vaginal discharge, postcoital bleeding, postmenopausal bleeding were included in the study. Pregnant women were excluded. Prior hysterectomy was excluded. Women with the clinical evidence of the acute pelvic infection was excluded from the study. Now, design of the work. Following this extraction method, the DNA extraction was done. The master mix was prepared following this master mix preparation chart. Now, thus the age-wise sample collection was done, but my study definitely aligned with the WHO, which says that from 31 to 50 age group should be the age group which should be diagnosed or screened for the cervical cancer. And my study definitely aligns. You can clearly see on the graph. This was the chart based on which we have interpreted the results. This is the RT-PCR graph showing the positive control, negative control, and the HPV. From among the 100 patients diagnosed with the HPV genotyping, 8 were overall positive for the HPV genotype, 16 for HPV 16, only 3 were positive. But the most prevalent were the other genotypes compared to the 16 and 18. And doctors are only focusing on 16 and 18 rather than going for the other genotypes. So I should definitely say doctors to. Okay. Now I have compared the pap smear with the RT-PCR. Detection is the cellular abnormalities here, HPV viral DNA. Genotype identification is not possible, here it is possible. Specificity, accuracy, and precision is very low in the pap smear, and in RT-PCR it is very high. So actually it is not moving. Molecular docking. This is my molecular docking results. I have done targeted molecular docking as the 2026 era is of the personalized medicine and the precision medicine. So we have given the targeted therapy for the genotyping. Like for 31, we got the strongest binding 45. For 52 and 58, we got the good binding, which is a good ADME analysis, which can be taken for the further experimental validations, and the biological efficacy can be checked, and further we can go for the clinical trials. Next. This is the heat map showing the binding affinity. The SP31 shows the strongest binding, and 52 and 58 shows the lead molecule. It is the lead molecule, which can be taken for the further drug analysis. The screening cost. Screening cost per test in India. Pap smear cost for 900 to 2500. Cervix shield, which I have designed, cost for only 1500 to 2500, giving you all the 14 genotypes. Other labs like Thyrocare and Apollo gives 4000 to 4500, which is costlier compared to my cervix shield. Now the global requirement of the kit is 130 to 150 million kits, and the gap is of 90 plus million kits are required. And in India, 25 million kits are required, and the gap is of 15 to 20 million kits. Definitely, I can align with some of the biotech companies, and definitely I can serve India for this gap for the 15 to 20 million kits. And a comparison of the cost, commercial kits are available at 1 to 1.10 lakh, and cervix sheet is available only at 50 to 55,000. Definitely 50% lower than the market available kits. Now conclusion, impact aligned with the WHO. Cervix Shield supports the WHO 90-70-90 cervical cancer elimination, which says that vaccination, screening, and target and therapy treatment should be given to all the women. Molecular docking identifies the scientifically justified lead molecules with for the targeted therapy. As we all know, the normal as we all know, the chemotherapy, in chemotherapy, 20 to 30% of the normal cells are killed, normal dividing cells are killed, but targeted therapy will definitely give you personalized medicine and will be saving your normal cells. Affordable kits, accurate and timely diagnosis reduces the cervical cancer mortality, extended HRSP genotyping, enables early, precise, and risk stratification. Targeted screening and targeted treatment will improve the survival outcomes for the women in India. Thank you.
Fine, thank you. So your presentation is over. Now, any single question? We can take a single question quickly, if at all is there. Any questions? Fine. Then we can shift to the next one. Next code is OA42. OA42. Who is OA42? Okay. OA43. All are requested to please follow the timelines. Only 5 minutes are given to you, as you know, and the last 1 minute will be given for the question and answers. Please follow the timelines. Complete your presentation within 5 minutes. After 4 minutes, you will be informed.
Good afternoon, everyone. Myself Sagar Dande, and today I am going to present my research work, uh, bioprospecting of rhizospheric soil for inulina's production, a steps towards sustainable enzyme technology and green biotechnology. So first, we need to understand what is the global context. Today, global need rising due to the bioethanol and high fructose syrup, and due to this, industrial enzymes are costly. So we need to eco-friendly and local alternatives. So let us understand what is inulin. Inulin is a natural polysaccharide which is found in plants like chicory, dahlia, and Jerusalem artichoke. And this inulin is important for various food and pharmaceutical industries. Now understand what is inulinase. Inulinase is a enzyme that hydrolyzes this inulin into fructose and fructooligosaccharides. And this inulinase is very important in high fructose syrup production, fructooligosaccharides, bioethanol production, and prebiotic and pharmaceutical applications. Now let us, I found these research gaps during the literature review. So I found this there is a limited exploration of rhizospheric microorganism, as well as inadequate studies are performed on these microorganisms. I will also find that there is a low enzyme and high production cost in commercial process, and limited optimization studies are also performed. So my objective is very simple and clear, to explore the rhizospheric soil for inulinase production, as well as to evaluate their role in sustainable and biotechnological applications. So let us discuss about how and where I conducted this study. I conducted this study at Department of Biotechnology, Government of Institute. Firstly, I collected the soil samples from various regions of Chhatrapati Sambhajinagar, especially from rhizospheric plants like banana and sugarcane. So after collection of soil sample, we designed, so these are the images of banana field and sugarcane field, and these are the images of samples that I have collected from the different region. After soil sample collection, we design a systematic methodology. This methodology consists of 5 major phases. In first phase is the sampling, which I have collected the soil sample from various regions. After that, I perform the isolation and screening method. Isolation is performed by stick plate and spirit plate technique. And then screening is performed with the help of Lugol's iodine method. After that, my third phase is a characterization of is microbial isolate. For characterization, we perform catalase test, gram staining, and indol test, followed by the extraction and purification of these enzymes. I partially purified these enzymes with the help of 80% ammonium sulfate. And lastly, we perform the enzyme assay.
and analysis. Enzyme assay is performed with the help of DNSA as well as Fallin-Lowry method. So let us discuss about the results, what I get. I get 11 colonies that shows the clear zone of hydrolysis by using a Lugol's iodine method. Out of these 11 colonies, I selected 2 colonies for further characterization and further studies. As we see clearly seen here, there is a growth of microbes on inulin media, and these are the results of biochemical characterization with the help of Lugol's iodine. So these are the results of biochemical characterization. I perform gram staining catalyst test and indol test. With the help of this test, we characterize these microbes with the help of their morphological and biochemical characterization. And lastly, we perform the partial purification of enzymes. So these are the results of Fallin-Lowry method. In Fallin-Lowry method, we estimate the protein concentration about 300 kgml. Okay, sir. So these are the results of DNSA assay. In DNSA assay, we found that the linear correlation is about 0.98, as well as we found that sample 1 has less concentration as compared to sample 2, which is a 0.30. Then let us discuss about how I can calculate the specific activity of this enzyme. I found that there is a 0.37% spatial activity of crude and 1.17 of purified enzyme. And this is a 3.18% fold purification. And I get 191% yield production. This is due to the removal of inhibitory products during the purification. So these are the industrial applications of my research work. These enzymes can be used in biochemical production, fructose syrup production, agricultural wells, valorization, as well as environmental benefits. So this is a conclude. I conclude that this rhizospheric soil is a great source of rhizospheric inulinase enzymes, as well as isolation, screening, and characterization perform their inulinase production activity. So these are the future direction. In future, I will study that there is scale-up studies, kinetic studies, then industrial applications testing. I sincerely gratitude to Dr. Rahul Bhagat sir, as well as all departmental faculty and students. These are the references that supports my research work. Thank you.
Okay. Any quick questions? Any questions from audience? Okay.
See, I have one question. What is the criteria for selecting these 2 plants only?
Sir, there is a microbial diversity of these rhizospheric plants. Other plants also consist of rhizospheric microorganisms, but these 2 plants are a greater microbial diversity.
So whether your endounulase which is being produced, whether it is the exogenous or endogenous product?
Both, sir. They generally consist of exoinulase as well as endoinulase.
Okay, fine.
Thank you.
Okay, fine. Thank you. Next, OFO 4.
Afternoon, everybody. Am I audible?
Yes.
I am doing my research under the guidance of Dr. Rahul Bhagat, sir. And my topic of research is cloning and expression of carboxypeptidase B in Escherichia coli. I have selected carboxypeptidase because I am working with insulin analog production at Wokhard Research Center. So I know what is the importance of carboxypeptidase B enzymes, because it is a pancreatic metalloenzyme that removes basic residues, lysine and arginine, from peptide C terminal, aiding digestion of the peptide. It is a zymogen. In our human body, during the production of insulin, insulin is produced as pre-proinsulin. It is converted by trypsin. But once trypsin has cut, it leaves back 2 basic amino acids, like basically arginine and lysine. So to remove this arginine and lysine, this enzyme we have to use it. And for cloning, we cannot clone. It is a 35 Dalton enzyme, but we cannot clone exactly 35 Dalton, because if we clone it, we don't get activity. So we have to clone it as a zymogen. It is a not pro-carboxypeptidase, but I have cloned as pre-pro-carboxypeptidase. It is a metalloenzyme. It needs zinc for its activity. So why have I selected E. coli? There are multiple expression systems, like you have yeast, you have mammalians, but I've selected yeast E. coli because faster doubling time, very good expression under the T7 DNA uh, polymerase. So, and we can get high amount of inclusion bodies expressed in it, and which will be, these inclusion bodies are much more pure than extracellular enzymes, and we don't have post-translational modification with carboxypeptidase. That is why I have gone with E. coli. And in E. coli, I have selected PET9A vector. Why PET9A? Because in I have two vectors with me, PET9A and PET3A. The drawback of PET3A is it has an ampicillin resistant marker and it is in the line. So sometimes we get run over transcription and ampicillin is produced, and then we cannot get better productivity. That is why, and PET9A I have kanamycin. That is why I have selected PET9A. And here you have T7 DNA polymerase, and IPTG induction can be given to it. So this is the amino acid sequence of the pro-carboxypeptidase that I have used. I have synthesized it from, I have given, synthesized it from outside this gene, and then I have cloned it into PET9A under NDA and BAMH1. This is the map of PET9A plus human carboxypeptidase. These are all the materials and components that I used, and this is the expression level. I got 9 to 10 colonies after transformation, after cloning and transformation. I got the clone from the outside vendor under PTZ57R, from where I have cleaved it with ND in bandmass and cloned it in PET9A and then transformed it in DL21DE3. Initially, I got it in DH5 alpha. When I expressed in 100 ml media and I induced it with IPTG, I only selected 3 clones. So these are the expression levels which I have got. There are, in the image you cannot see, there are a lot of impurities up and down, but I have got good expression level. And the first band, it is a 47.
Last 1 minute.
Yeah, the first band to the left, what we can see, it is a standard 47 kilodalton protein, known protein, and these three are of my expressed protein. So the molecular weight is 46.87 Dalton, and the theoretical PI, which I will need in future for purification of this enzyme. So this is it, and I can use it. There are many applications for this enzymes, like you can see in research aid and industrial process protein engineering. But my main aim is to use it in insulin production. Now, at present, before 2 to 3 years, this is not 2 to 3 years, before 10 years, when I was procuring this enzyme was nearly 3 lakh rupees a gram. It is used in insulin. It is used in fast-acting insulin Lispro. It is used in fast-acting insulin Aspart. It is used in insulin Lispro. It is not used in glargine. In this, it is used. And even in the upcoming projects like Degludec. In Degludec, we take insulin sequence, cut it, and then put a fatty acid chain on it. So my main aim is to use it in insulin process.
Thank you. Any questions? Okay, thank you. OFO 5.
ऐसा होल्ड करो.
Good afternoon, everybody. My name is Kailas Lipne, and I'm a research scholar in Government Institute of Science. And my research topic is enhanced hyaluronic acid synthesis in recombinant E. coli strain through genetic engineering. So I'm doing this research in Wokhart Research Center. So first of all, we can first of all, I would like to tell you the what hyaluronic acid is. So hyaluronic acid is a polysaccharide. It's a chain of two dipeptides, two like a beta glucuronide acid and beta acetyl glucosamine. There is a linkage actually, so that would be repeated continuously till the 1000 kilodaltons. Also, it's a high molecular weight molecule. Also, there are small molecular weight molecules also are there. So what it helps in the joint in our joints. So it maintain the hydrophobic condition in our body, and it is also used in some of the thermal products. It's a very good product for osteoarthritis also, to maintain the joint lubricancy, and it reduces the pain in joints. It reduces the injury in joints. So this is overall, its molecule is very important for us, and we can say this is molecular weight of there are three types of molecular weight observed in hyaluronic acid. That is high molecular weight, medium molecular weight, and low molecular weight. So as per the needs, they will produce this hyaluronic acid in the industry. So how this hyaluronic acid is produced? So naturally, this hyaluronic acid is produced in Streptococcus species. So all the Streptococcus species can produce this hyaluronic acid. This is the pathway actually. It's a glucose dependent pathway. So when the Streptococcus will be there, will be like so this ABC transport will transport the glucose to the inside the cell. Then that there would be two pathways, the PNP pathway and glucose glycolysis pathway. So out of that two pathways, there would be two important precursor molecule will be produced. One is the glucuronic acid, and another is an acetyl glucosamine. So there are some enzymes also involved into the convergence. So all enzymes are related to some of the genes also, but there is one gene, that is hyaluronic acid synthase A. That is the last enzyme which is important for linking these two precursor molecules, that is glucuronic acid and N-acetyl glucosamine. So this enzyme is present in Streptococcus, so it's like naturally get produced in the Streptococcus. So it will be isolated, and it will be it will be used in the for the pharmaceutical products, and so in the cosmetic products. So this is the polymerization reaction. That last reaction is very important for this conversion. So my topic of interest is hyaluronic acid synthesis by recombinant E. coli strain. As we know, that why do we need to do this? Already it is synthesized in the Streptococcus. So why? So E. coli. So my interest is E. coli because if we see the Streptococcus is first of all, it's a pathogenic organism, and another is it will produce the endotoxins while processed. So these are the two important aspects of any process in the industrial products. When you are using this product into pharmaceutical interest, so we need to have this criteria. There should not be endotoxin, there should not be pathogenicity. So to reduce that pathogenous effect and all the process, I want to transfer this process into E. coli. So there is the pathway will be same in the E. coli also, like Streptococcus. There's only one difference into that is last hyaluronic acid synthase one enzyme, A enzyme, that is responsible for connecting two precursor molecules. It is not available inside the E. coli.
Conclude. Last 1 minute.
So key important step is like selection of hyaluronic acid synthase A, then cloning into a vector, that is that I have cloned it into a PET28 vector, and this is the gene sequence that I took from NCBI, get synthesized from gene script. That these genes, this vector will be produced, and this vector will be transferred on PL21. So it is a E. coli, so it will automatically produce the hyaluronic acid once this enzyme is getting synthesized into this. So approximate production yield will be here is we can expect 1 gram, 1.5 gram, up to 1.5 gram in batch feed for E. coli, and at the commercial scale that will be 2.5 or 3 gram, up to 3 gram per liter. So in the Streptococcus, it is giving more yield, but there are some disadvantages also. That's why I'm shifting this to E. coli. So yield wise, we can see recombinant E. coli can produce high yield. If you see applications, as I said, osteoarthritis therapy also will be having the more response for this product. Then dermal fillers, like it's like depends on the molecular weight, this would be used for that. Then wound healing capacity is there. So final outcome of this product is the enhanced HA production in E. coli. Then that is a novel strain will be produced, sustainable production because of the pathogenicity removal, and high quality hyaluronic acid can be produced into the project. So challenges would be this, that would be metabolic changes also, metabolic engineering also can be done, and some CRISPR-Cas technology also can be used. These are some challenges into this, so we can improve the strain improvement. Thank you.
Any questions?
Whether you had synthesized this hyaluronic acid in the E. coli?
Sir, it cannot be. Yeah, I'm doing, but not yet synthesized, because till now I have completed gene synthesis.
See, I would like to know whether you had some result with you.
No,
or not.
not yet completed.
So it's just a hypothetical.
No, till half. Because references available, it's not hypothetical.
What are your findings? What are your findings?
Till now, I didn't get any results because I'm in the middle of the research, because I have completed clone development. So I'll do the further transformation, and then I'll get the yield. So after getting a transformation, I can do some media optimization by glucose feeding and all. Then hyaluronic acid can be synthesized into the E. coli. So that I will present in.
So how you are sure that you had closed the proper organism and the gene has been transferred?
So gene transformation can be confirmed by there is.
No, I am talking about what research you are working now right now.
Right now, there are two objectives, like cloning of the genes, hyaluronic acid synthesis into a E. coli, and second aspect is like.
So whether you had succeeded in any of these objectives?
Yeah, first objective.
Okay. Yes.
Okay, thank you.
Next is OFO6. Since you have a time of just 4 minutes, so focus more on your material methods and the result discussion.
Okay, good afternoon first of all. My presentation topic is GCMS analysis of N-hexane extract of Clitiva adansonine DC with insight into molecular pharmacological mechanism. So first of all, Clitiva adansonine is a small tree or plant. It has ethnomedicinal properties belonging to Caparidaceae family, and it is a widespread use of leaves, bark, and as well as the root of this plant has led to increase in pharmaceutical as well as phytochemical research into this plant. There are ethnomedical significances. Clitiva adansonine used in multi-purpose topical ethnomedicine for anti-inflammatory, anti-rheumatic, anti-diabetic, analgesic, and wound healing application. So, in anti-inflammatory activity, leaf extract reduce pro-inflammatory cytokines such as TNF alpha, then interleukin 6, interleukin 8, then anti-parasitic or anti-malarial activity, in which preliminary in vitro anti-plasmodium strain, that is Plasmodium bergii in mice. Here are some applications. Next, so my objective is to perform phytochemical profiling to extract using GCMS chromatography, mass spectrometry, that is GCMS, to identify and characterize the major volatile and semi-volatile compound which is present in that extract, to identify compound via NIST spectral matching, to correlate the identified compounds with reported pharmacological significances or activities, to explore possible molecular mechanism underlying the biological potential of the detected phytoconstituent. So here is a material method collected of mature plant leaf of Clitiva adansonine, and then dry in shade. After that, we grind it, and after that, in N-hexane solvent, we oxalate and then send to the sample MIT college for further process, that is GCMS. Here the information of GCMS, which equipment is used, what is the column size, and which gas is used, that is helium gas is used. Here is the GCMS analysis. GCMS analysis of N-hexane extract developed 30 compounds. After that, here are the TLC chromatograms show retention time. Uh, Here is the list of 30 compounds which we have get. So which one of them is 1,3 oxidized derivatives, in which these compounds with nitrogen heterocyclic reported antimicrobial, anti-inflammatory, and anti-cancerous activity. Then thiourean L, that is 1,1-dimethyl L-phenyl thiourea, that is known as antimicrobial and enzyme activity. One of them compound is that is quinoline. Why I selected this quinoline? Because it is synthetic drugs and it is anti-cancerous drug. Quinoline derivative identified in N exam of extract of radioadenine belonging to quinoline class of heterocyclic compound. Quinoline derivatives are widely reported in research as potential anti-cancerous agent. They also show antimicrobial, antiparasitic activity as well as enzyme inhibitory activities. So how quinoline work or proposed mechanism of quinoline? First of all, induction of programmed cell death, that is apoptosis. First of all, quinoline derivative increase ROS, that is reactive oxygen species, and inside cancer cell. Then ROS generation disrupt the mitochondrial membrane potential, and when mitochondrial membrane potential is disturbed, cytochrome C is released, and in the cytoplasm or into the cytoplasm, is triggering the capsase cascades. That is the capsase activation of Cas9, and which is further activated by 3 and lead to apoptosis. Second, cell cycle arrest. They can block progression at G2M checkpoint, preventing cancer cell division.
Concluding last 1 minute.
1 minute. No?
Yeah.
After that, signaling pathway, that is modulation. Quinoline derivates the many suppressed survival pathways such as PIK3, MAPK, and ERK, enhancing anti-cancerous activity. And last one, that is topoisomerase inhibition. Some quinoline compound which inhibit DNA topoisomerase enzyme, causing DNA damage and cell death. Last line, that is quinoline derivatives are reported to be active against multiple cancers including breast cancer, lung cancer, colon cancer by inducing apoptosis and inhibiting cell proliferation. Here are my conclusions. GCMS analysis revealed the presence of multiple bioactive compounds including quinoline and triazine derivative along with phenolic and fatty acid ester compounds. Next, that is quinoline triazine scaffolds are widely reported in the literature of anti-cancerous activity, including apoptosis, induction, and cell cycle arrest. The second last, the presence of pharmacological important chemical classes suggest potential therapeutic significances of the plant extract. For last objective, that is further in vitro and in vivo studies are required to scientifically validate their anti-cancer potential. Here are references and thank you.
Fine, thank you. Any questions? Okay. Apart from sending samples for the GC, what exactly the experimental work you did?
I have done SOCSLET at the Department of Botany, Government Institute of Science. After that, when report came, then I have found review literature of that compound.
That's what I said. Experimental work. Apart from sending samples for the GC, what is the other experimental work you did?
No, sir. Collection of plant sample and SOCSLET.
See, in your GCMS analysis,
yes, sir,
which are the additional compound other than the earlier people who had discovered?
Sir, in which nitrogen heterocyclic compound are there, fatty acid are there, then 1,4-dio-nitro something there, fatty acid is there, then heterocyclic compound, and list of 30 compound, and 1 or 2 are contamination of that machine, that is GCMS. There are not there.
Okay, fine, thank you.
How this quinoline gonna stop the checkpoints in cell cycle, like for progression of cancer?
Actually, I have not done this work in lab or in vitro condition. It is based on review or literature. Thank you.
OA47. Next is OA47.
Afternoon, one and all. Myself, Aishwarya Kishore Kulkarni from Department of Microbiology, Government Institute of Science. I'm here today to present my research work on green synthesis of silver nanoparticle and its application as nanofertilizers. We all know that agriculture plays an immense important role in growth and development of a country. But about 30 to 40% of agriculture sector is dependent upon synthetic fertilizers for the crop production. Hence, here comes the role of nanotechnology. Now, what is nanotechnology? Nanotechnology simply means technology at nanoscale. Hence, the peculiar characteristic of nanofertilizer that high surface to volume ratio provides deeper penetration, effective stability, and also provides higher crop yield due to slow release mechanism. Now, the main mechanism of the nanotechnology, as I have earlier said, that high surface to volume ratio. Now, the difficulty arises in the synthesis of nanofertilizers because of the extreme optimal conditions required for it. Achieving eco-friendly, high-yield, cost-effective, and sustainable production of nanoparticles is a significant hurdle. Hence, my research addresses all those problems, and I'm here with the synthesis of neem leaf extract and chlorophyll mediated synthesis of silver nanoparticles, and its applications are studied in terms of nanofertilizers in which I have studied its impact on wheat seed germination particularly. In order, for, in order to analyze a comparative study, chemical synthesis method was also performed parallelly in which TVA served as the capping agent and trisodium citrate served as the reducing agent. Here is the literature review, which says that green synthesis, that is biological synthesis of nanoparticles, is safer alternative to the chemical synthesis, as naturally the plants have the flavonoids, terpenoids, glycolides, alkaloids, phenolics, etc., which serve naturally as a reducing and capping agent. Several researchers have obtained high yields in improvements in maize, sunflower, and fenugreek seedlings because application of silver nanoparticles has positive effect on the plant growth and also improve the biochemical content as well as morphological parameters. Here are the objectives. First, green synthesis of silver nanoparticle. Second, characterization of nanoparticle using instrumented methods. Third, application of green synthesized silver nanoparticles as nanofertilizer. And fourth, a comparative study of green synthesized and chemically synthesized nanoparticles. The method of synthesis of nanoparticles, silver nanoparticles, mainly I have categorized according to the three ways which I have performed. The first one is neem leaf extract mediated synthesis of silver nanoparticles, in which I have used 20 ml of the leaf extract combined with 80 ml of silver nitrate, then further centrifuge for 5000 rpm for 10 minutes, and the dried pellet was used for further absorbance measurement. Next, chlorophyll mediated synthesis of silver nanoparticles, in which 30 ml of chlorophyll solution was added to 100 ml of...
1 minute. Last 1 minute. You just have to conclude in 1 minute.
Okay, sir. Fine. And the third one is the chemical synthesis, which is the traditional method for the synthesis of silver nanoparticles. The characterization of synthesized nanoparticles was carried out by UV visible spectroscopy. The effect of seed germination was calculated by the germination percentage. And the third, plant growth parameter was analyzed by like sugar content, and a protein content was analyzed by DNSA and Biorate method respectively. The chlorophyll and carotenoid content was optimized and calculated by Arnon and Davis method. These are the results. The figure 1 shows the successful synthesis of silver nanoparticles, seeming the color change. The second figure illustrates the successful synthesis of silver nanoparticle as the peak is obtained in the range of 420 to 450 nanometer. And the third figure illustrates the growth parameters in terms of shoot length of the wheat seeds. Here are the results of morphological analysis and biochemical analysis, which significantly illustrates that.
It's over.
Okay, sir, thank you.
Just conclude in 1 to 2 minutes.
The green synthesized silver nanoparticles effectively enhance the growth parameters in terms of morphological, that is seed germination, shoot length, root length, and the biochemical analysis like sugar content, protein content, and chlorophyll and carotenoid contents was extremely increased as compared to the chemically synthesized, which showed a toxic effect on the seed seed germination and other parameters. Thank you.
Thank you. Any questions?
In the control, the value are similar.
The germination percentage was similar. The 100% germination was obtained in control as well as green synthesized both methods, but the chemically synthesized silver nanoparticles showed a toxic effect. But as we are seeing the other parameters, it has showed a significant increase in other parameters like sugar, chlorophyll, carotenoid, and protein levels. Okay. Thank you, sir.
Thank you. Next is OFO8. OFO8. OFO9. Yes, yes. It will be better if you can focus more on material methods and result discussion.
Good afternoon, everyone. Today I will present our research topic, plant-mediated silver nanoparticles in synthesis of the silver nanoparticles and their antimicrobial and photocompatibility.
Keep your mic closer to your mouth.
Next. Our our main studies focuses on the green synthesis of the silver nanoparticles by using the pepper betel leaf extract and their evaluation of their environmental and biological applications. So nanotechnology is the study of the nanoparticles which have size range of 1 to 100 nanometers and which have properties, chemical, physical and biological properties. Nanoparticles have inorganic, organic and carbon-based materials. Inorganic part contain various metal and metal oxides. Organic part contain various plant extract and biomolecules. So in the inorganic part, we may use the various metals like gold, silver and copper. Here we have used silver for the synthesis of the nanoparticle because it has antimicrobial activity and various silver nanoparticles have a great role in the biomedical, environmental bioremediation, biosensor and antimicrobial coatings. So silver nanoparticles is the suitable metal we used for the nanoparticle synthesis. The conventional method for the nanoparticle synthesis were used toxic chemical like sodium borohydride, which causes the environmental hazard and which also causes the high energy consumption. So to overcome this problem, we used green synthesis like biological source for the synthesis. We have used a plant extract for the synthesis of the nanoparticles. We have used a pepper betel leaf extract for the synthesis because it have various phytochemicals like flavonoids, polyphenols, tannins, and alkaloids, which have a great role in stabilizing the nanoparticles, and it is also act as reducing agent and capping agent. Reducing agent means it reduces the monocationic silver ions into the silver metal for the synthesis of the nanoparticles. And it also acts as a capping agent, means it stabilizes the nanoparticles. Along with it also prevent the agglomeration of the metal ions. Means in the inorganic part of the nanoparticles, various metals have been used which form the agglomerization. So it prevent the agglomerization. So here we have used the paper betel, so it will act as a capping agent and reducing agent, which also have the medicinal value like an anti-inflammatory agent. It also prevent the wound healing and gastrointestinal. So it act as an ideal source for the synthesis of the nanoparticles. Objective to synthesize silver nanoparticles using eco-friendly green route via paper betel leaf extract. We have used a paper betel plant extract along with the silver night solution. The color change in the mixture observed from pale yellow to brown, which evaluate means analyze that nanoparticle synthesis was confirmed. To perform the phytochemical screening of the extract to identify reducing and stabilizing agent. The phytochemical screening means presence of the phytochemicals, presence of the carbohydrates, proteins, alkaloids, which gives the identity that the reducing and capping reagents are present in the nanoparticles. To characterize the synthesized silver nanoparticles and identify functional group using a FAIR spectroscopy. In the FTIR, we have confirmed the various functional groups, which also confirm that the cyanide group, amine group, hydroxyl group, and keto group.
Yes. Conclude in 1 minute. Quickly.
So I will hand over the materials method to my colleagues Kajal.
Anyways, you have to conclude in 1 minute, better.
Okay. As per the conclusion, so here we prepared the plant extract of paper petal leaf, and here we prepared the here we done the powder form of the paper petal leaf, and we prepared we use as a AgNO3 as a precursor, and we kept that mixture under the dark condition. Here we perform the phytochemical analysis. According to the phytochemical, it shows that there is a flavonoid carbonyl group is present in the according to the phytochemical analysis, and we explore the potential applications. Here we focus on the antimicrobial activity as well as photocatalytic dye degradation. So, according to the FTR, there is a carbonyl amino group as well as aromatic amino group is present. Majorly it is present as the metal oxide, which is responsible for the dye degradation. Here we perform the antimicrobial activity. The efficiency of antimicrobial activity is majorly shown against the Klebsiella.
The time is over. Just conclude in 1 or 2 lines. Quickly.
Okay. Overall, we will conclude that successfully demonstrate that that paper betel leaf has the strong efficient activity against the human pathogen, as well as it shows the strong activity as a photocatalytic dye degradation.
Fine. Thank you. Any questions? Yes. Yes. Please come here.
If we are talking about the antimicrobial activity of silver nanoparticles, what do you think that by which mechanism they are killing the bacteria?
Actually, what happens that when that silver nanoparticle comes under the contact with the visible light, that time it goes under the excitation. Due to the excitation, it enters into the it forms the radical hydroxyl radicals. That hydroxyl radical responsible for the degradation of azo dye or the azo bonding present in the that azo dye or that complex. So it is responsible to chelate with that molecule.
How you authenticate the material, plant material?
Sir, it is under the process. We submit to the university.
Fine. Thank you. Next is OA410.
A very good afternoon to one and all. So, the title of my research is the screening of the bacteriogenic lactic acid bacteria from floral microbiomes and the optimization of their product. So, in short, why have we selected floral microbiomes as a source? Because they are rich in sugars and amino acids, and thus they will harbor lactic acid bacteria, and we are studying the potential of their bacteriocin production. So this is the methodology in short. First, the collection of flour samples, followed by isolation and screening of bacteriocin producing lactic acid bacteria, then exploring the antimicrobial potential of the lactic acid bacteria, and lastly followed by the bacteriocin production optimization by employing statistical methodology. Now, methodology for isolation and screening. We had utilized MRS medium for isolation. Around 40 different flour samples were selected, from which 50 different isolates were obtained. And utilizing biochemical tests, 18 were to be confirmed as the lactic acid bacteria. Further, the bacteriocin producing potential was checked by bacteriocin assay, which is the agar-well diffusion assay, and the selfie supernatant, abbreviated as the CFS, and neutralized selfie supernatant NCFS of all the isolates were tested so as to decipher whether the antimicrobial effect is due to the H2O2 or due to the proteinaceous bacteriocin. Positive control was used as a positive control of the standard microorganism L-lactis MTCC 440. The indicator stain was used as Micrococcus luteus MTCC 106. And lastly, the presence of bacteriocin was confirmed by its proteinaceous nature was confirmed by treating it with various peptide enzymes. This is the methodology for bacteriocin production optimization. Firstly, suitable ranges, they were recognized by one variable at a time approach. Then the factors, which factors were significant, it was studied through Plackett-Berman, and the important or significant factors were then further optimized by using the response surface methodology. The results of the antimicrobial effect of the CFS, all the 18 isolates, they exhibited antimicrobial effect, but upon neutralization, the neutralized selfie supernatant, only one isolate, which was CC1, isolated from cucumber flower, it showed activity against Micrococcus luteus. These are the zone of inhibitions performed in triplicates with standard deviation. And as you can see, all the isolates showed inhibition. Only the neutralized selfie supernatant of the isolate CC1 exhibited antimicrobial effect against Micrococcus luteus. Then its neutralized selfie supernatant was treated with proteinase K, and as desired, upon treatment with proteinase K, the bacteriocin loses its activity, confirming its proteinaceous nature. Effect of different enzymes from the bacteriocin. So different enzymes, all the peptidases, as expected, it neutralized the effect of the bacteriocin, proteinase K, trypsin, and pepsin, whereas catalase did not affect the bacteriocin's efficiency, which means that the antimicrobial effect is not a result as of the hydrogen peroxide. Also, amylase did not affect its production, means there is no carbohydrate moiety involved in the inhibition. Then susceptibility of different indicator strains, not just Micrococcus luteus, but also Listeria monocytogenes, which is also a deadly pathogen. It is also inhibited by this bacteriocin. Apart from that, Lactobacillus plantarum, which is a major food spoilage agent, it is also inhibited by this particular fruit bacteriocin. Molecular identification of the isolate was identified as L-lactis by performing the 16S rRNA gene sequencing as well as by BLAST, and the sequence was submitted to the NCBI database and the accession number was obtained. OVAT results, pH 7 was ideal for both the production and growth. Temperature of 30 degrees was suitable for both production, whereas if we see in the graph, higher temperatures result in no production.
Last 1 minute, conclude.
Plackett-Berman analysis showed that temperature, inoculum, peptone, and glucose were the major components involved. These are the results of Plackett-Berman analysis. Response surface methodology and the contour plots. The central regions and the curve nature shows that the results are significant with a p-value of 0.001. This is the verification of the analysis that the optimization was successfully done. It was also checked in triplicate. Growth and bacteriocin production profile. The growth was maximum at 18 hours, and after 24 hours, the bacteriocin production stabilizes and it remains thereafter. These are the outcomes. Novelty of the research work is that floral microbiomes are the least explored in terms of the lactic acid bacteria, And also L-lactis. This is the first instance in literature review that L-lactis has been discovered in cucumber flowers.
Thank you. Any questions? Any questions from the audience? What is the significance of your research work?
So the significance of this research work is that bacteriocins are yet only used in food industries, but they also have a potential to eradicate the current problem of the antibiotic resistance, as they can be used as substitutes towards the antibiotics.
For antimicrobial activity, positive and negative controls were used.
Yes.
So which organisms use for positive and negative control?
Sir, for positive control, the industrial strain, which was obtained from MTCC, which was the same L-lactis, but it was derived from cheese. So it was used, it's self-supernated and neutralized self-supernatants were used as positive.
But its source is different, no?
Yeah. But it is only the standard microorganism, and it is officially recognized as Nizine producer. So that's why we are using it as a positive control.
Fine, thank you.
Fine, thank you. Next is OFO 11.
Hello. Good afternoon, one and all present over here. There is an ancient saying in the Sanskrit literature that is Yatha Pinde Tatha Brahmande, which means as in the atom, so is in the universe. So in the 21st century, where we see that the nanotechnology is a booming area, we are using nanotechnology in different aspects of the agricultural field. So this nanoscale has to be studied in various aspects so as to get its correct application in the field of the agriculture while taking a step towards the informed nano agriculture. So the introducing the part of my research work is, so we say that the copper oxide nanoparticles, the copper is a micronutrient for the plant growth. We are using copper oxide nanoparticles as a biofertilizers as well as the biopesticides. But if we are using them, there is a bound interaction which is going to be happen between the nanoparticles which we are using and the PGPR, that is plant growth promoting rhizobacteria. So in this study, we are going to assess the effects of copper oxide nanoparticles against the PGPR. So these were the objective of my study. The first one is we were using Aspergillus flavius for synthesizing the nanoparticles, copper oxide nanoparticles, by using a green approach. Once the synthesis was done, we wanted to characterize the nanoparticle, physiochemical characterization, in order to get to know their size, shape, and stability. And upon thorough characterization, we went for the toxicity assessment against the rhizospheric microbial community. So this is the mycosynthesis of copper oxide nanoparticles by using Aspergillus flavius. As you can see in the flow chart, we can clearly see that we have used the cell-free filtrate of the Aspergillus flavius. Alongside with that, the Aspergillus flavius and the L-ascorbic acid both act as the reducing agent, while the copper sulfate pentahydrate acts as the metal precursors. Upon mixing all of them, followed by the photoexcitation, we see a color change from the colorless to the yellow color, followed by the red precipitation formation at the bottom of the vial, confirming the visual synthesis of the copper oxide nanoparticle. Once the visual confirmation was done, we went for the characterization of them. At first, we went for the UV visible spectroscopy, where we can see that the LSPR peak, that is local surface plasma resonance peak, was found to be as the 634 nanometer, which is specific for the copper oxide nanometer. FTR showed there is a amide 1 and amide 2 linkages, which shows the fungal protein capping is present on the copper oxide nanoparticle, which is stabilizing them. Zeta potential was recorded as the minus 9.6 millivolt, which further says that the nanoparticles which we have synthesized are moderately stable. And the DLS analysis shows there is a bimodal distribution of the nanoparticles. The TAME analysis is the key characterization which we have used, as it shows the morphology of a synthesized nanoparticle, which we can see that is acicular, that is needle-like shape, and the NTA, which shows the hydrodynamic size of the nanoparticle, shows average size of the 38 nanometer in the liquid solution based on the Brownian motions. Once the nanoparticles were thoroughly characterized, we went for their effect on two selected microorganisms. One was this Bacillus thuringiensis and second one is Pseudomonas fluorescens. First one is a very well-known biopesticide, and the second one is the plant growth promoting rhizobacteria. As you can see in the graph, we can see that the Bacillus thuringiensis shows maximum sensitivity at all concentration. That means the BT is most sensitive to the copper oxide nanoparticles comparing to the Pseudomonas fluorescens, which shows resistance up to the 100 microgram per ml concentration. So this shows that over here we can see that the higher concentration of copper nanoparticles, they are lethal for the Pseudomonas fluorescens, as well as whereas the lower concentration of the copper oxide nanoparticles can inhibit the growth of the BT. Further, we evaluated the overall toxicity of these copper oxide nanoparticles against the overall rhizospheric microflora, where we can see there is a yes, sir, where we can see there is a depletion as we go, increasing the concentration of the nanoparticles, which suggests the dose-dependent toxic effect of the copper oxide nanoparticles against the microflora which is there in the rhizospheric soil. You can see in the control, the percent inhibition in the control where the soil is treated with the distilled water only is 99.5. But when we go, pardon for my mistake, the control shows the 0% inhibition, but when we increase the concentration, that is 150 microgram, we can see that 99.5% of the inhibition occurs, indicating total functional sterility of the rhizospheric soil. Concluding, we should know that the copper oxide nanoparticle should be used below or equal to the 75 microgram per ml of concentration, which is a threshold limit for the uses of copper oxide nanoparticle. Beyond that, they are showing toxic effect. And the second one is, if we are going to use generically copper oxide nanoparticles, they are going to be having the toxic effect on the PGPR. But if you are using their moderate use, we are going to be promoting effect of the PGPR. Finally, acknowledging my guide, Dr. I.R. Gupta sir, and the Department of Biotechnology, Government Institute of Science.
Fine. Okay.
Now I am open for discussion.
Thank you. Any questions? Okay.
Thank you, sir.
Next is OFO12.
Hello, am I audible? Good evening, everyone. My name is Sangram. I'm presenting my work. So the topic of my research is marine algal diversity from the coast of Murud. So this is the abstract, which shows that we found out we found out accurately 20 species from the coast of Murud, which we collected from 11 different locations along the Murud coastline. So this is the introductory part. As we know, Indian coastline is very rich in for marine algal resources. We have almost 624 algal species recorded, and for the Raigad district, from the review, from the research review, we found out that 45 species are found in Raigad coast. In Murud Tehsil, in our research, we collected 20 algal species from the 11 different locations. Here is the slide of our steps. First of all, here is a map. The map of India magnifies into the map of Maharashtra, and here you can see the our coast side, Murud. So this is what we did. We collected samples of the from the location. We process it, we rinse it, we wash it with the sea water and the fresh water. After that, we preserve the samples in wet and dry format. For the dry format, we did the herbarium preparation method. And for the wet, we made a solution of 4% formalin. The 4% formalin we did for the maintain the cellular structure of the species. So here you can see the collection site, and there is habitat and collected material. These are the species we found on the different locations. I will be share that in observation. These are the member of Phaeophyta. You can see the Colpominia, Padaina, Sargassum. We found out 3 Sargassum species from these research. After that, we found out Spathoglossum and Dictyota. These are some members of Chlorophyta. We found out these species, Ulvas, 2 species, Colorepa, 2 Entomorpha, Chytomorpha, Brachis, and Entomorpha compressor. So these are some members of Rhodophyta that we found out. Gracilaria, 3 species of Gracilaria, 1 Giladium, Amphifora, and Lifora. This is how we preserved, and this is how we labeled our species. Sample name, location, the coordinates of the location, the date, and collected by. So this is our wet preservation. This is how dry preservation looks like. These are the herbarium, and same the name of species, locations, coordinates, date, and collected. So here are my observation. From the coast of Karle, which is a village near from Murud, from the Karle, we found out 2 species from of Sargassum, which are Tenerium and Illifolia, which rather its application is for biofertilizers and alginate. From the coast of Murud, we found out total 3 species of Sargassum: Spathoglossum, Colpominia, Dictyota, and Ulva. So these are the different kind of applications which are taken from the reviews of literature. There, this species Sargassum is used for agar and alginate. Pathoglossum is used for biofertilization. Pulpominia, Dictyota, and Ulva is an edible seaweed. So in the coast of Bhandarwada, Sargassum, 1 species, and 1 species of Pathoglossum we found out. Okay.
Last 1 minute.
Yeah, yes, sir. So here are different remaining locations: Vihur, Donator, Nandgaon, Turumdi, and Rajapur. And these are the species. So these are the locations. This is how it looks. Yep. This is our conclusion that we found out different species from the different locations. We found out almost 6 species from Rhodophyta, 7 are of Chlorophyta, and remaining 7 are of Phaeophyta. So this is the compilation of the diagram. 35% Phaeophyta, 35% Chlorophyta, and 30% of Rhodophyta were found on our collection. These are my references. Thanks.
Fine, thank you. Any questions? Any questions from the audience? So can you tell me what is the significance of your research?
So sir, it was a checklist to check in to the course that how many species are there. Like it is the diversity survey kind of thing. And after that, we are into the some bioactive biochemical research as well. Thank you, sir.
Next and the last number is OA413. OA413.
Hello, everybody. Thank you for patiently listening and all the presenters who have presented here. With this last presentation, we have concluded this session, and immediately we will be having the last technical session followed by the valedictory. So you have to be present here till the end. So I request your uh, attention as well as your presence till the end. Thank you.
लाल कलर का फोन यहाँ रह गया, यहाँ से आके कलेक्ट कर। कौन सा? कौन सा? वाटर।
जहाँ पे चल रहा है सेशन वहीं पर फोन भी है ना। जी ऑडिटोरियम में जहाँ लैपटॉप रखा है ना। जहाँ से प्रोजेक्शन हो रहा है।
यहाँ बैटरी है।
थैंक यू।
ठीक है।
इसका है क्या 50% है। शुरू में 60 पे लाया था 10% तो सिर्फ
चार्जिंग पे लगा लेना।
नहीं,
इसका
लगा
बैटरी
था।
बैकअप अच्छा है।
हाँ 18 घंटे चलता है।
बढ़िया है।
मेरे चलता 15 मिनट।
हैं 18 मिनट।
क्या?
इसका लैपटॉप 18 मिनट चलता है।
इतना भी नहीं।
कौन सा आपका?
HP। ये नहीं, ये तो सर का है। ये अच्छा था।
अरे भाई टाइम भी हो गया है रे, उसको 10 साल हो गए उस लैपटॉप
ये अभी 10 साल चलेगा, अभी ले आऊंगा 10 साल।
उसे 10 साल हो चुके
कहाँ
हैं।
जा रहा है? टेक्निकल सेशन कब है अगला?
बस होने वाला है।
खत्म करवाओ।
पोस्टर पे क्या है कुछ?
आया
मैं भी आती हूँ देखूँ।
आपको वापस देना पड़ेगा ना, यहाँ रख दे रहा यहाँ रख रहा है एक को।
Hello, good afternoon. We are moving into the next technical session. So I would request all of you to remain calm and quiet and be attentive to what will happen next. Uh, we have a technical session whose resource person is Dr. Kishore Gawai. So he has already graced this auditorium. So I would request him to move on to the stage. I also request Dr. A.N. Salve, who will be the coordinator for this session. I request him to take over the proceedings. And I also request chair of today's technical session, Dr. Nitin Adapure, to also move on to the podium.
So, very good evening to one and all present here. Friends, we are moving in the second last session of this one-day national seminar on the recent trends in biotechnology. For this session, we have a eminent speaker, Dr. Gawale sir, and I would like to request Bhagat sir from the forensic science to kindly visit Gawale sir, please. Bhagat sir. Just Adapura, sir. Okay. Okay. I would like to request Bhagat sir to kindly felicitate the chairman of this session, Dr. Nitin Adapure sir. Take this opportunity to kindly introduce today's chairman, Dr. Nitin Adapure, who is the at the head of biotechnology and microbiology and zoology of the Vivekanand Sardar Arts, Dilip Singh Commerce and Science College, Chhatrapati Bhajanagar. Sir has worked for teaching experience of 15 years as well as the research experience of the 15 years. To his credit, there is one novel species of the fungi, that is Orobacidium primulum, which he has published in a journal with an impact factor 11.65. To his credit, there are a couple of research projects, almost six projects, five projects are there, and the total grant is 55 lakh 81,000 he has transferred. He has transferred some of the technologies, particularly the low-cost biogas storage balloon system. At the same time, he has developed the e-waste management system, then laboratory waste to bioenergy production, and for this he has been the patent has been granted to him, and one new patent he has filed for that. He is also having some consultancy work, and almost 68,000 rupees has been generated by the consultancy. And he is collaborating working with several agencies also. Very big biodata is there. It will be very difficult to read all these things. Almost he had authored 14 book chapters, books, and almost 9 book chapters also. So very huge biodata is there. Total research students, one research fellow is working with him. 5 research students are working under the funded project that is by DBT, BIRAC and DST, Government of India. On this occasion, I would like to request sir to work as a chairperson of this session. Over to you, sir. Please.
Thank you, Dr. Save sir. So, for this today's session, we have a speaker, Dr. Kishore Gavade sir, with us. So let me take an opportunity to introduce Dr. Gavade sir. So Dr. Gavade sir is currently working as an assistant chemical analyzer DNA division in the Regional Forensic Science Laboratory, Chhatrapati Sambhajinagar. He has a vast experience in carbohydrate chemistry and synthesized 4 to 6 membered imino sugars as glycoside inhibitors. He has also synthesized some sugar-based peptides and studied their conformations. Basically, he has acquired a master's degree in organic chemistry from Dr. Babasaheb Ambedkar Marathwada University. Further, he has qualified GET and CSR NET, and he acquired his PhD in organic chemistry in 2017 from Savitribai Phule Pune University. Further, he has developed certain skills in organic-based automated synthesis, automated extraction of the DNA, and even he has acquired skills in the QR-PCR, then the GA of forensic data samples. Further, he has also developed some skills in organic, that is a multi-step and solution phase peptide synthesis. He has delivered lectures, invited talks in various conferences. Further, he has attended various seminars, workshops, and conferences. He has a work experience, particularly as a senior research fellow in R&D division in MKO Pharmaceutical, Pune, and he has many research publications in his credit. So it's an honor to introduce and to welcome our today's speaker, Dr. Kishore Gavade sir. So welcome, sir, and I request Gavade sir to please start your presentation. Thank you, sir. Yeah, before that, I would like to felicitate Dr. Gavade sir on behalf of the conference. So I would like to request Dr. Anjali Naik ma'am to felicitate Dr. Gavade sir, please. Thank you, ma'am. Thank you, ma'am. So I would like to request Dr. Gavade sir to please start his presentation.
युवराज स्लाइड्स बंद कर सर कॉलर माइक पॉइंटर देखिए लाइव ये कॉलर माइक हां उबे पॉइंटर दे सर हो सर दे तुम शो लो चांगलं चालतं हे लगा दे
Hello. Okay. Pointer.
It's working. ठीक आहे का हां बरोबर काय म्हटलं सर अजून का मला कम्फर्टेबल Can we put on that light, please? That light. I think it's not visible. That light. No? Okay. Let it be. Good afternoon, everyone. I am Dr. Kishore Suresh Gavade, working as an assistant chemical analyzer in DNA division of Regional Forensic Science Laboratory, Chhatrapati Sambhajinagar. First of all, I would like to thank the organizers, for inviting me to this national conference on recent advances in biotechnology for health, agriculture, environment, and forensic sciences. I would also like to thank my Deputy Director, Sri Narendra Gosavi sir, who allowed me here to represent RFSL Chhatrapati Sambhajinagar for this prestigious event. Today, I'll be talking on applications of biotechnology in forensic sciences. Before going to the actual talk, I would like to give a brief overview of forensic science laboratories across Maharashtra. We have total 8 regional labs located at Mumbai, Nagpur, Pune, Nashik, Chhatrapati Sambhajinagar, Amravati, Nanded, Kolhapur. We also have mini labs at Dhule, Ratnagiri, Solapur, Thane, and Chandrapur. Our head office is located at Mumbai. There are various divisions in which the analysis is being carried out, such as toxicology, biology and serology, DNA, general analytical and instrumentation, prohibition and excise, cyber forensics, tape authentication and speaker identification, ballistics, physics, psychology, and wildlife. In today's talk, I'll be giving a brief introduction of biotechnology and forensic sciences and the various techniques which are used currently in the DNA forensic sciences. And in the end, we'll have a question and answer session. Biotechnology is a multidisciplinary field that has emerged by combining Sorry? Biotechnology is a multidisciplinary field that has emerged by combining biology with technology to create innovative solutions and advancements in various domains. It integrates expertise and techniques from various fields such as molecular biology, chemistry, biochemistry, chemical engineering, and digital computing. On account of its unique ability to serve and benefit humanity, the last 2 decades have seen unprecedented growth in biotechnology. Biotechnology has its immense application in forensic science too. Forensic science uses science and technology for the detection and investigation of crime. Accordingly, forensic science laboratories provide an unbiased scientific opinion and help the judiciary based on the scientific analysis of evidential materials provided by the respective investigation agencies. Biotechnology has revolutionized forensic science, providing powerful tools and techniques for analyzing biological evidence, identifying individuals, and reconstructing crime scenes with unprecedented accuracy. Advances in biotechnology, ranging from genomics to proteomics, have significantly increased the accuracy and efficiency of forensic investigations. Its applications span a wide range of spectrum, from DNA profiling to advanced protein analysis. This has fundamentally transformed how the criminal investigations are conducted and how the justice is being served. One of the pioneering applications of biotechnology in DNA forensic analysis is DNA fingerprinting by restriction fragment length polymorphism. This was developed by the British geneticist Sir Alex Jeffreys. In this technique, specific regions of DNA surrounded by variable number of tandem repeats are cut into fragments by restriction enzymes such as endonuclease. These DNA fragments are then analyzed with the help of gel electrophoresis and subsequent probing and hybridization technique to get the DNA fingerprint. So apart from DNA fingerprinting, this technique is also used in genetic mapping and identification of genetic variations. This technique is quite expensive and requires large amount of high quality DNA. Hence, currently this technique is obsolete and replaced by STR analysis, that is short tandem repeat analysis. This is one of the most impactful biotechnological application used in the forensic DNA analysis, which is DNA fingerprinting by short tandem repeat analysis. This technique relies on analyzing highly variable regions of the human genome called short tandem repeats. These are also called as microsatellites. So what are these STRs? These STRs are short repeating sequences of nitrogen bases in DNA. They typically range from 2 to 6 base pairs in length, and that vary in length between individual to individual. The analysis of these variations forms the basis of DNA fingerprinting, which gives it a high discrimination power. Now, I would like to give the brief overview of steps which are involved in the DNA profiling using STR analysis. In our laboratory, particularly in DNA division, we receive various kinds of cases, such as paternity, sexual offense cases, homicidal cases, cases of identification in case of missing person, concealment of birth cases, and cattle cases. In cattle cases, different types of meat samples are submitted to our laboratory. There, we find out the origin of that meat sample and also give the gender of that sample. Whether that sample belongs to cow or buffalo, there we that kind of report we give in our laboratory. So, depending upon the type of cases, our sample can be any biological material. So, when the sample is blood or a part of aborted tissue, aborted fetus, there is no problem for sampling. We directly proceed for DNA extraction. But sometimes we receive samples like product of conception. Here you can see there is a large amount of mother's uterine tissue along with the developing fetus tissue. So here the sampling becomes critical. For such kind of samples, we prefer random method of sampling and select 2 to 3 tissue sample from these various portions and analyze each of them separately. This is one of the exhibit which we have received recently in our laboratory. This was a case of concealment of birth case where the abortion was carried out illegally and the fetal remains were buried in the farm, which was wrapped in the sanitary pad. This was brought to us by the respective investigation agencies. So for such kind of samples also, we prefer trial and error method so that we choose selective portion and go for DNA extraction. Once the sampling is done, the next step of DNA analysis is the DNA extraction. So DNA extraction can be performed by 2 methods. First is organic method, the second one is automated extraction method. In organic method, various chemical reagents are added manually and it requires largest sample quantity. As the chemical reagents are added manually, it increases the chances of contamination and sample degradation. So to avoid this, most of the laboratory now prefer automated method of extraction, which works on pre-programmed extraction protocols. These extraction machines just look like robotic platforms. So they provide increased efficiency as well as speed. So as the machine seems to be a robot, it reduces the risk of contamination and sample degradation. Along with that, it also ensures standardization of the procedure. DNA extraction process involves lysis of cells to release the DNA molecules, then separation of the DNA molecules from other cellular material, finally, the isolation of the DNA. At our laboratory, we have all these advanced instruments which are used for the DNA extraction. For example, EZ1 and EZ2. These are from Kaijing and Automate Express, which is manufactured by Thermo Fisher Scientific. EZ1 and Automate Express, in these machines, we can process 15 samples at a time. In the advanced version, EZ2, we can process 24 samples at a time. So once the DNA is extracted and is in hand, the next step in the DNA analysis is quantification of that DNA by real-time polymerase chain reaction. The primary purpose of DNA quantification is to determine the appropriate amount of DNA template to include in the downstream process, that is PCR amplification of STR loci. Presently, as little as 0.5 nanogram to 1 nanogram of DNA is sufficient to generate the DNA profile with the current commercial STR kits. I am telling you that this amplification process needs optimum amount of DNA. So why I am telling you this? If you can see in this figure, if we proceed with larger quantity of DNA, it will give poor results. You can see in the figure A. Or if you have too much little DNA, then it will give false result. You can see allele dropout here. So when the DNA quantity is appropriate, we'll get such a nice DNA profile you can see in the figure C. This quantification is done by using RT-PCR machines. We have these two machines at our laboratory, 7500 and the advanced version, Con Studio 5. So once the exact amount of DNA present in the given sample is determined, the next step in the DNA analysis is amplification by polymerase chain reaction. This polymerase chain reaction was first described by Kary-Mullis in 1985. And for this outstanding discovery, he won the Nobel Prize in Chemistry in 1993. First and foremost, we all know that near about 99.9% of DNA of all human is same. Remaining 0.1% of DNA, which is different in every individual, makes it unique. So these STR loci present in this 0.1% of DNA region. These are selectively amplified using sequence-specific fluorescent primers, making millions to billions copies of the particular DNA sequence in shorter amount of time, which are necessary for further analysis. This amplification process is carried out with this thermal cycler, which has 96 wells, meaning it can process 96 samples at a time. So this PCR step, it involves three major steps. In the first step, the double stranded DNA molecule is converted into single strand by denaturation. In the next step, that is annealing, here the fluorescent labeled primers get attached to the single strand of DNA. And in the third step, which is elongation, DNA elongation, this elongation step is carried out with the help of polymerase enzyme along with DNTPs to get the DNA molecule. So in the first step, one DNA molecule is converted into two. And in the next step, two gets converted into four, and so on. So at the end of 30 cycles, millions to billions copies of particular DNA sequence are produced in a short amount of time. So once this amplification process is done, the next step is genetic analysis. So genetic analysis is carried out in a special instrument called genetic analyzer. This instrument basically works on the principle of capillary electrophoresis. Firstly, the samples are placed into a tray, which is placed here and injected into the capillaries. The capillaries are located here, and a high voltage is then applied. As we all know, DNA molecule possess phosphate groups on their backbone. So, under the action of electric field, these DNA molecule migrate from cathode to anode and gets analyzed. So, here you can see the cathode buffer container is placed here and the anode buffer container is placed here. So, under the action of electric field, these DNA molecules migrate from this position to here. And here the detection window is there. Here their analysis is being carried out and we get the results. So fluorescently dial labeled PCR products are separated and analyzed as they pass by the detection window, which is placed here. Better resolution between closely sized DNA molecules is achieved as the electrophoretic separation is carried out under denaturing condition. All the steps, including injection, separation, and detection, are fully automated. So this permits analysis of multiple samples in a shorter amount of time. This is one of the instrument 3500. We have both the versions of this instrument, that is 8 capillary and 24 capillary, meaning 8 capillary can process 8 samples at a time. 24 capillary instrument can process 24 samples at a time. So once the genetic analysis is done, we get the DNA profile by using gene mapper IDX software. The DNA profile looks like this. You can see the different STR markers over here and the respective allele calls, maybe homozygous or heterozygous, depending on whether this particular individual has inherited same or different alleles from his parents. As we all know, every individual inherits its DNA from his parents. So DNA of every human being is unique, and no two persons can have the same DNA profile except monozygotic twins. After generating the DNA profile, the next step comes the interpretation of the results. For the sake of understanding, I have taken 2 examples. Here you can see the first case to your left. This was a paternity case where we have received the 3 blood samples of mother, baby, and father. Here you can see the genotype of the baby. The baby, you can see has inherited one allele either from his parents. At the first locus, you can see he has inherited 15 allele from father and 16 from mother. So after comparison, you can see mother and father matches all the maternal and paternal alleles respectively at all these STR loci. After matching this at all STR loci, then we can say this particular baby belongs to this pair of parents. In another case, you can see the genotype table of this case. This was a case of sexual offense where a semen stain was found on the victim's cloth. The DNA profile was generated. Here you can see. Then the blood sample of the accused was submitted to our laboratory, and the DNA profile of that accused was also generated. And here you can see it was matching with the accused. So this matching is done at all STR loci, and you can see if it matches, then we can say the DNA profiles obtained from these 2 exhibits are identical and they belong to the same male origin of that person. So during our interpretation, we face some challenges such as mutations. Mutations are alterations in DNA sequence. These are caused by errors in DNA replication, faulty repair mechanism, or because of environmental damage to the DNA. Here you can see it was a case of paternity analysis. This female child has shown mutation at this particular locus D13S317. Here you can see this female child is homozygous, showing allele called 8. So she has inherited allele 8 from her mother and allele 8 2 from her father. But you can see the father is homozygous with the allele called 13. So this is an example of mutation, which is 5-step mutation. Instead of inheriting 13, the baby has inherited allele 8. Sometimes we face wide deletion of, in some of the male profiles. This occurs because of mutations during spermatogenesis or maybe because missing genes on Y chromosome. So whenever the DNA profiling is done, the DNA profile of such kind of sample which belong to male, it primarily looks like a female DNA profile, because you can see at the amelogenetic locus the Y amplification is absent. So only X is amplified. So it sometimes create confusion. But additional Y-STR analysis gives us the proper idea. Another type of challenge sometimes we face in our analysis is the inheritance, which is a triallelic inheritance. This I will explain in the next slide, because this triallelic pattern I have observed in one of my case when I was in the regional forensic science laboratory in Nagpur. It was a case of paternity testing. Blood samples of mother, alleged father, and the product of conception were received to our laboratory for DNA testing. After the thorough analysis, that is DNA extraction, quantification, then PCR, and finally genetic analysis, we found that father and mother matched respective paternal and maternal alleles present in POC sample at all STR loci, and then were concluded to be the biological parents. Here you can see the triallelic pattern. Here this POC sample has inherited allele 16 from its mother. Here you can see, and both the alleles from its father. So instead of appearing as a homozygous or heterozygous, it appears as a 3-band allele pattern. So this was observed at as many as 4 loci, along with a mutation at this locus. So this was one of the interesting case, and this observation we have recently published in one of the Springer Nature's journal recently. These triallelic patterns arises because of somatic mutations, localized intrachromosomal duplication, or maybe because of chromosomal aneuploidy. Another application of biotechnology in forensic DNA analysis is YSTR analysis. This YSTR analysis is a highly accurate male-specific DNA profiling method focusing on short tandem repeats on the Y chromosome. As the Y chromosome does not undergo recombination, it is passed down intact, because of which all males in a direct paternal line share the same YSTR profile. This analysis is particularly useful in detecting male DNA in samples where there is a large amount of female DNA. For example, genital swabs of female in case of sexual offenses. It is also used to trace patrilineal ancestry and for kinship or paternity testing. It becomes more useful in resolving identification cases when the sample of father is unavailable. As I said earlier, the male belonging to same family share same YSTR profile. This technique becomes useful for such kind of cases. This technique has some limitations too. While excellent for inclusion, it has lower discriminatory power compared to autosomal STR analysis, meaning it cannot easily distinguish between closely related males, for example, father and son. If father and son are both accused in the same offense, so on the basis of only YSTR, we cannot distinguish that whether the father is involved or the son is involved, because both will be giving the same YSTR profile on YSTR analysis. So only on the basis of YSTR analysis we cannot distinguish uh, you know the accused in such type of cases. Sorry. Another application of biotechnology in forensic DNA analysis is mitochondrial DNA analysis. Mitochondrial DNA, which is inherited maternally, is present in higher copy numbers than nuclear DNA. This makes it valuable for analyzing highly degraded or ancient samples such as bones, teeth, where nuclear DNA is present in very small amount. So this mitochondrial DNA is circular in nature, and the sequencing of the hypervariable regions such as HVR1 and HVR2 of mitochondrial DNA helps in comparison of maternal relatives. Wildlife DNA analysis is another application of biotechnology in forensics. So, wildlife DNA analysis is conducted to identify unknown wild species from parts or remains of animals that are generally unidentifiable from their morphological characters. The principle on which DNA analysis works here are same, but wildlife DNA analysis has its own challenges. The species identification of the samples is done using mitochondrial DNA, while gender determination is done using nuclear DNA. The sequence of mitochondrial DNA thus generated is submitted to the basic local alignment search tool, which is also called as BLAST. And this is developed and maintained by the National Center for Biotechnology Information. This BLAST identifies unknown species by comparing a sequence from the known species against a large database of known genetic sequences. This wildlife analysis, this facility is available at Regional Forensic Science Laboratory, Nagpur. One of the advanced application of biotechnology in forensics is next generation sequencing, which is also called as high throughput sequencing. It's a modern, rapid and cost-effective method used to determine the exact order of nucleotides present within the DNA or RNA molecules. Unlike older methods such as Sanger sequencing, this NGS utilizes massively parallel sequencing to analyze millions of fragments simultaneously. So this enables sequencing of whole genomes or specific genes in short amount of time. Therefore, NGS has largely replaced older and slower technologies like Sanger sequencing for large-scale projects, allowing for more comprehensive understanding of complex genetic data. Its uses include identifying genetic variations such as mutations, single nucleotide polymorphisms, disease diagnosis, especially in case of cancer, pathogen identification, and evolutionary studies. Another application is proteomics in forensic sciences. Proteomics, which is the study of proteins, nowadays gaining increasing importance in forensic sciences, particularly in forensic medicine. So, analysis of these protein expression patterns provide valuable information about an individual's physiological state or exposure to certain substances. There are various techniques employed in the protein analysis, such as mass spectrometry, enzyme-linked immunosorbent assay, Western blotting, etc. So this has found application in body fluid identification, time of death and estimation in cases of post-mortem interval, in toxicology, and in species of origin identification. So these were the recent and advanced techniques and applications of biotechnology in forensic sciences. Uh, there are opportunities for all of you students to in come and work in state as well as central forensic science laboratories. And you also have opportunity to work in national forensic science universities as a scientific assistant, assistant chemical analyzer, or students from cyber specialization, they can work as scientific officers. One thing I would like to suggest to forensic science students is that you go through chemistry. You read chemistry books. You make your chemistry stronger. Then and then your chances of getting selected will be increased. Because, in the last exam, I have seen out of the total candidates who were selected, most of them, around 80 to 90%, were from chemistry background. And only 20 to 20% students were from forensic background. If you make your subject chemistry stronger, you will strengthen your candidature for this examination. So I will urge you to go through chemistry. Now the session is open for questions. If you have any questions, you can ask.
Yes, any questions? Feel free to ask questions. There are so many students over here from forensic subject as well as from biotechnology subject. So if you have any queries, any small doubts, you can ask. Yes?
Yes.
Good evening, sir. Sir, I have a very curious question for you. We always hear about the paternity disputes
Yes.
that generally happen. So, for that, which methods are used, like DNA fingerprinting and all we hear about?
Yes.
And I would also like to know about the accuracy of the results of these tests. Sir.
Okay.
Thank you.
I have explained this in one of my slides that DNA fingerprinting method is the only method for testing the paternity of particular child, and the results which are coming, these are very firm results. There is no challenge to these results. And what was your next question?
Other methods.
Other methods, generally all the laboratories now perform DNA fingerprinting for paternity analysis in cases of this paternity testing. So there are hardly any other method for this testing.
Yeah, excuse me, sir. One simple question I want is, in this slide, there is STR locus that you have listed.
Yes.
So is this STR locus mandated in forensic to be used, that this specific 25 set that you have displayed, this should be used, or each lab has their own individual choice which to compare in a particular test that they are doing? So frequently they go back to do that same test, or randomly they can pick up any loci and compare and just give the analysis. Is there any particular number and choice?
In the ancient days, when the DNA profiling was started in the 1980s, there were only 5 to 7 loci. Scientists, developed. After that, scientists developed many loci. The vast amount of study was, performed from starting from 5 locus. Now we have almost 23 to 27 loci to study. So, there is no doubt if start with 5 locus and give your analysis. If that matches, that gives confirmed results. But yeah, it also depends on the type of kit which we are using. So nowadays, all the companies are manufacturing about 23 to 27 markers kits. And in the nowadays, the standard practice is you should have at least 20 markers to give your results. So according to SWATM, 20 Codiscore STR loci are preferred for DNA analysis. Yes. If there are no more questions, thank you. Thank you for your kind attention.
लाइट चालू करो तो पीछे 3 स्विच ऑन करा इधर पीछे साइड में इधर पीछे की साइड जाएगा ना 3 स्विच है
Thank you, Dr. Kishore Gawle sir, for your nice and lucid lecture and drive us into the role of biotechnology in the forensic science. On this occasion, I would like to invite the chairman of the session to give his remarks.
Thank you, sir. So thank you, Dr. Gawle sir, for this such a fantastic lecture. And in this lecture, he has actually given us a journey to complete DNA fingerprinting, RFLP, SDR, then the triallelic pattern, mitochondrial DNA analysis, wildlife DNA analysis, NGS, and at the last, the opportunities in the forensic science labs. Even in his deliberations, he has actually talked about some cases such as the paternity cases, sexual assaults, then the concealment of the birth. So as I know, like there are so many students over here who are from the forensic science, forensic biology, as well as biotechnology. So like all of these things you have studied in your textbooks, or we can say in your theory lectures, as well as some of this you have also performed as a practicals. But see, he is the person, Dr. Gawde is a person who is working on this at the ground level, so that he has explained it with the certain examples. So like the actual samples, say for example the DNA extraction and RFLP and all, so definitely you have performed such experiments in your lab. But the actual samples he is using at his lab, and he's doing certainly a great work over there. One of the important point which I have pointed out is, like he has also focused that so many students who have qualified this particular examination, like the scientific assistant, assistant chemical analyzer, and the scientific officer, so they are having a strong chemistry. And I don't know whether you have also pointed out the thing or not. Like I have introduced himself. Like he is a person from organic chemistry, and he continuously he's talking about all the applications of biotechnology in forensic sciences. So this is like he's a certain example in front of you. A person from a chemistry, how fantastically he is doing work in the biotechnology, and that's what like whatever we have heard about like the interdisciplinary work, interdisciplinary research. What is this? So this is nothing but the same thing. So that's what we have to focus on. And here again, I would like to point out one more important thing, which I have heard just a few days back. Like whatever the recently the Nobels we have got, the Nobels in biology, I'm saying. So these persons, they are basically from chemistry. So they have used their chemistry knowledge and they have applied it in the biology. And like that, they have got a Nobel Prize. So this is what we just have to think about. We are a person from biology, but I don't know how much strong our chemistry is. So the same thing he is also saying, he is also emphasizing that we should strong our chemistry as well. So that's what you can see the takeaway message from his talk. So once again, I would like to thank Dr. Gavre sir for this fantastic talk. Thank you so much.
So we are at the last stage of this technical session. That is a vote of thanks. It's my pleasure to present my thanks to the speaker, Kishore Gowle sir, for giving his valuable time and remarkable note, particularly on the role of biotechnology in the forensic science. As well as I would like to thank to the chairman of this session, Dr. Nitin Athapure sir, for chairing the session. At the same time, I would like to thanks to the organizer for giving an opportunity and thank you. With the kind permission of the chair, I declare that this session is over. Now we are going to assemble here in the same lab for the valedictorian. Thank you.
ले लो लगे होते पेंड्राइव आ गया पेंड्राइव आ गया
आ गया
ओके लगा दो कल ऊपर
म्यूट करके चलाना जब लेकर तो वरना सब खड़े हो जाएंगे।
चल चल
नहीं
चल चल जब तक ये लगा देंगे तो मैं आग भंग रहा है या भी कुछ
वैलिडिक्ट्री
क्या
वैलिडिक्ट्री तो ये ऊपर कितनी चेयर बैठने वाले वरना ये वापस रखना पड़ेगा बस कम रखते बेचारों पूछना पड़ेगा
इससे ज़्यादा तो नहीं होंगे शायद से
बस कर दो
हाँ कर दो
Good afternoon, everyone. The valedictory function is going to start in few minutes.
आवाज नहीं बनती कोई भी आवाज बनती
वहाँ पे कुछ तो कर सकते रहे थे ऐसे सुबह से तेरा अकाउंट है तेरा अकाउंट है सुबह से 5:30 घंटे चलाएंगे
पहले 1 घंटा
मोबाइल मर जाएगा ना और बैटरी खत्म हो गई ना उसकी चार्ज नहीं बैटरी खत्म है सर
चालू है क्या 5 घंटे कितने घंटे दिया 5:30 से पहले 1 तास 1 तास वो वाला
पता है क्या डायरेक्ट सर की बैटरी कम से कम 10% तो भी गई होगी
10% होगा तो कुठला उलट केला चार्जिंग
नाही रे 10% असं नाही बॅटरीची लाईफ 100% असते ना ते डायरेक्ट 90 झाली असते डायरेक्ट 90 हा नाही
मेरे फोन में भी है स्पीड भी अच्छी मैं घाट बात नहीं कर रहा सुबह से माहिती शिवानी बात कर ये कांड बात कर रहा है तो तेरे को भाई ये जो है ये 1 मिनट का बस एक मिनट ठीक है 1 मिनट ज़्यादा हो गया अरे अरे सफिशिएंट है इतना तो होना चाहिए ना सुना देखो नहीं मतलब
मैं अपना अपना लगा लेंगे फिर आगे बढ़ेंगे
1 मिनट सफिशिएंट है
क्या नींद कहीं दे लो कहीं नहीं रे मजाक कर रहा मजाक कर रहा पागल
मेरे पास कौन कितना
रुषा भी
होगा उसने लिखा ही बोलेगा
सुनना रुषा भी बोला कि मेरे पास काही नाही फॉर्म काढा जात कानामध्ये काही नाही फॉर्म काढा जात बोर अरे वो दूसरा लगा ना बाबा
दूसरा काय लावू
नाही भी लगा
दूसरे लोग कोणते मध्ये
विनोद क्या लगा ना विनोद सेंड कर ना
मध्ये ऍड वगैरे आली ना तर पैसे दिला पडेल
त्याच्यामुळे व्हॉट्सअपला डाऊन ऑफलाईन कर
ते ऑफलाईन आहे
ते व्हॉट्सअपवर बोलून घेईन त्याच्याकडून डाउनलोड
हे नॅशनल अँथमला येईल आपल्याला काय
जय जय महाराज
तो पण इसको डिस्कनेक्ट करके सुना मेरे को वाला 1 मिनट का ये
लगा बोला जाऊ दे ऐकून तर दे ना बाबा त्याला नहीं नहीं लगा लगा इस पे भी ये 5 सेकंड के लिए तो लगाना क्या
नहीं लगाना जाने दो ना है
मैं लाइव चल रहा यूट्यूब पे लाइव चल रहा मेरे अकाउंट पे
कसं युट्यूबवर करतात काय करतात युट्यूबवरती गेले 5 घंट्यापासून चालू आहे 5 घंट्यापासून 5:30 काही नाही पाहणारे फक्त 4 जण येतात काही टीचर सांगतो 4 जण पाहते रोज प्रचार असतो लावायचं लावून सर लोक डायरेक्ट युट्यूबवरती लाईव्ह करा तुम्ही पाहिलं तर दिसेल सर्च केलं ना आपण डॉक्टर चरण सिंग काटे लाईव्ह अच्छा त्यांच्या स्वतःवर केले शाळेच्या जागी 5 दाखवलं चांगलं मोठं चॅनल झाले हे सगळं त्यांच्या त्याच्यावर करतो माझं पण काय करतो तू माझं बायोटेक्नॉलॉजी लास्ट इयर करा चांगलं चांगला स्कोप आहे असे काम आमच्याकडे
शिकायला भेटायचं इथला इतिहास आहे मोठा या संस्थेचा नाही ना मी त्यात बसत तुम्हाला बसायचं हा चालेल त्याला करावं लागतं ठेवा 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 धन्यवाद हो हो ओके सर बाकी कुठले प्रॉपर मी सेलूच आहे सेलू मानवत अच्छा हे आपले टोपे साहेब माननीय आता कोण आहे तिथं झालं टोपे साहेब बरेच वर्ष होतं भाजपचं सीट आहे ना आता भाजपचं आहे ना शेलूला आमचे बरेच नाते मानस शेलू ना चांगलं गाव जावं पण तिथेच आहे नाते बरेच नाते संभाजीनगर सगळ्यात चांगलं आहे तिथला प्रवास एक तासानंतर कार्यक्रमात आहे ना मी मला सांगितलं एक तासानंतर मी कार्यक्रमात आहे हा तिथं ऑनलाईन काम चालू आहे संभाजीनगर मुळगाव इकडेच आहे 35 वर्ष हे डिपार्टमेंट इथलं काम माझ्याकडे जवळपास 25 वर्षापासून 25 वर्ष मी विषय लोक आमच्या वेळेस वर्षभरचं बॅकअप नसेल 30 वर्षाचं आमच्याकडे विद्यापीठ आहे गव्हर्नमेंट सगळे कॉलेज आहेत विद्यापीठ नाईक कॉलेज विद्यापीठात वगैरे युवक महोत्सव वगैरे सगळं आपण छापलेलं असतं लोकमतला वेळ तुम्हाला काय पाहिजे फोन करा या वाटल्यास मिळून देईल काय सर इथे कसं आहे इथे लवकर घेत नाहीत डिपार्टमेंट वाले बी घेत नाही यांना क्रेज नाही घेऊन त्या अवघड बिल देतात ते बिल सोडत नाही आधी पण ते बाकी विद्यार्थ्याला विद्यार्थी किंवा प्राध्यापक घ्यायची एवढी हे नाही कारण सगळे बिजी असतात एकदा गेले सगळे नाही नंतर देतात गव्हर्नमेंटनुसार असतं ते होतं जास्त येत नाही कुणी इकडे 5,000 पॅकेज आहे 5,000 100 फोटो आणि बाकी माझा मित्र पण आता फोटोग्राफीमध्ये ग्रामीण मध्ये चालते आता सिटीपेक्षा पहिलं सगळं राहिलं नाही आता मोबाईलमुळे बरंच काही संपले आता हे जर ऑनलाईन असतं तर 5,000 रुपये मिळाले असते हे तुम्हीच करायचं ना जुगाड आहे ना नाही असं सुरू झाले पायावर पाय झालेच नाही आता असं म्हणता बी येणार नाही आहे ते आहे नाही तर पहिलं एवढ्यासाठी आम्हाला बोलवायचे तुमचा लॅपटॉप घेऊन या आमच्याकडे नाही लॅपटॉप ते तुम्ही सांभाळा तुमचा आता कसं झालं तुमच्याकडे मोबाईल प्रत्येकाकडे लॅपटॉप झालाय आता घेतलेलं आहे माग दसऱ्याला घेतलंय ना कितीला घेतलं आयुष्यात पहिलं झालंय त्याला 10 वर्ष वापरण्यावर आता 10 वर्षापेक्षा कुठलंच नाही काही 20-25 लागलो एकदा नवीन घेतले टाकलं सकाळी नाही नवीनच आहे 80 मी घेतलं होतं त्यात एवढं काही कळत नाही मला सोनीचा कॅमेरा होता अल्फा सिरीजचा लेन्स हे मला वाटायचं कॅमेऱ्याची बॉडी महाग असेल लेन्स स्वस्त असेल उलट ना कॅमेरा भेटेल तुम्हाला लाख 1.5 लाख 2 लाख रेंजर 3 4 हे त्याची बॉडी 2.5 लाखाची बॉडी आहे लेन्स त्याच्यापेक्षा 350 हे हा कामाला नव्हता माझा कॅमेरा बाहेर गेला म्हणून आलो हे आणत बी नाही हे कंपनीसाठी हा कोणता कॅनॉन 60 मार्क एक ते रेड नावाचे कॅमेरे असतात शूटिंगसाठी सोनीच असतं सोनी नाही नाही रेड नावाची स्पेशल रेड हां रेड ते वायफाय वाले आहेत ते कनेक्ट करणे एकदा लावून दिले नेट वाले हो ना इंटरनेट त्याला हां हे वाले वायफाय कनेक्ट केले की संपले खूप चांगले असतात पण चालत नाही ना आपल्याकडे पैसे देणार लोक नाही आपल्याकडे पैसे कार्यक्रम आहे खरे जे लोक बिझनेस होता जैन मारवाडी दोघांचा खरा बिझनेस पाच लाख रुपये देत होते बरं का त्यांच्या धर्मगुरूंनी सांगितलं काहीच करायचं नाही विशेष नाही खाण्यावरचा खर्च कमी झाला बाकी समज नाही एवढं त्या समाजाचं 10 लाखापर्यंत जात होतो आम्ही काहीच म्हणत नाही 10 लाख आता ते कामच कमी झाले दोघांचे एकच कॅमेरा मुलींचे फोटो जास्त काढायचे मुलांचे कमी काढायचे सगळे असं करा हात करा तसं काय काहीच नाही काम केलं त्याला कॅमेरा तो जोडला का हा कॅमेरा केला का वेब कॅमेरा केला असं पासवर्ड टाकला असं आजकाल शूट करतात पा जी नवीन पोरं वगैरे आलेत ना नोकरी चालेल शिक्षणापासून दूर आहेत ना ते जे शिकायला इंजिनियर म्हणून आहेत ते सध्या फोटोग्राफर राहिले त्यांच्या आई वडिलांनी काय केलं एमजीएमला बघतो ना मी एमजीएमला 200 कोर्स सोडून टेक्निकल AI ला वगैरे शिकायचं त्यांचं ते सोडून इकडेच पळायचे लग्नात ह्याच्यात त्याच्यात त्यांनी आलेत कशासाठी आज तुम्हाला 2000 रुपये मिळाले काहीच नाही सोडून द्या वाटतंय तसं याच्यात पडल्याशी कळत नाही माणसाला 30 लाख रुपये याच्यात असं वाटतंय माणसाला त्याच्यात जॉब पडतो ना तुम्ही त्याच्यात पडता तेव्हा ते कळतंय माणसाला आधी छान वाटतंय हे सगळं आता दिवसभर मेहनत करतो 1 रुपया नाही मिळालं कळतं त्याला काय आम्ही 35 वर्ष झाले मी याच्यात 35 वर्ष मी जर जॉबला असतो तर माझ्या घर शांत राहिलं असतं त्याच्यात घर शिक्षण वगैरे याच्यात कसं आहे वेळ जास्त जातो आणि जसं जसं वय होते ना तसं तसं लोक दूर करते या फील्डमध्ये त्याची कमाई 25 ते 35 नंतर लोक वय विचारते समोर आता नवीन पिढी काय विचारते तो अंकल न्यायचे आहे मी माझ्या जवळच्या तर पुन्हा माझ्या मुलाला सुद्धा हात लावू देणार नाही सर 50,000 काही किंमत नाही काहीच किंमत नाही 10 वर्षानंतर तुम्ही विचारणार 1000 रुपये शक्य होते 50,000 मोबाईल वगैरे पण आले AI मुळे काय झालंय आधी तुम्ही जुने फोटो दिलेले आम्हाला क्रिएटिव्ह करायला पैसे घ्यायचे आम्ही आता काहीच नाही तू AI वर टाकले 2 मिनिटात माझी गरजच नाही तुम्हाला माइक ऑन है ना माइक
I welcome our honorable dignitaries. I request everyone to please stand up. I request honorable dignitaries to have a seat on dais. I also request convener of this conference, Dr. Mayawati Patil Madam, and co-convener, Dr. Anjali Naik Madam, to have a seat on dais.
पैसा this is all because of money.
Honorable Chief Patron, Shri Venugopal Reddy, Additional Chief Secretary, Higher and Technical Education, Government of Maharashtra, our patron, Honorable Dr. Shailendra Devrankar Sir, Director, Higher Education, Government of Maharashtra, respected Dr. Pankaja Vadhare Madam, Joint Director, Higher Education, Chhatrapati Sambhaji Nagar Region, respected dignitaries on dais, distinguished guests, faculties, research scholars, delegates, and my dear students in offline and online mode. I welcome you all to the valedictory function of National Conference on Recent Advances in Biotechnology for Health, Agriculture, Environment, and Forensic Sciences, RAB 2026, conducted in hybrid mode, organized by Department of Biotechnology, Government Institute of Science, Chhatrapati Sambhajinagar, in collaboration with Department of Forensic Biology, Government Institute of Forensic Science, Chhatrapati Sambhajinagar, under the aegis of Dr. Baba Saheb Ambedkar, Marathwada University, Chhatrapati Sambhajinagar. I would like to begin this valedictory ceremony with a timeless Sanskrit wisdom. Sa Vidyaya Vibhuktaya. True knowledge is that which liberates. Over the course of this conference, we have witnessed knowledge through keynote sessions, oral and poster presentation that has liberated. The theme of the National Conference, RAB 2026, beautifully reflects interdisciplinary scientific progress. We are honored to have Sri Narendra Gosavi Sir, Deputy Director, Directorate of Forensic Science, Maharashtra State Regional Forensic Science Laboratory, Home Department, Chhatrapati Sambhajinagar, as a chief guest for this valedictory function. And we also have guest of honor, Sri Abhijit Rashe Rao Sir, Assistant Director, Regional Forensic Science Laboratory, Chhatrapati Sambhajinagar. Dr. Ms. Sri Gaure Sir, our Honorable Director, Dr. Rajendra Satpute Sir, is the chairman of today's valedictory function. Dr. Mayawati Patil Madam, convener of conference, and Dr. Anjali Naik Madam, co-convener of this conference. Atiti Devo Bhava is our tradition. I request our Honorable Director, Dr. Rajendra Satpute Sir, to felicitate our chief guest, Sri Narendra Gosavi Sir, by presenting him with a shawl, shrifer, and bouquet. I again request our Director, Dr. Rajendra Satpute Sir, to felicitate our guest of honor, Sri Abhijit Rashe Rao Sir, Assistant Director, Regional Forensic Science Laboratory, Chhatrapati Sambhajinagar, by offering him a shawl, slipper, and bouquet. I request Dr. Rajendra Satpathy sir to welcome Mr. Gaure sir by offering him a bouquet. And to felicitate again with a shawl, shreefer and bouquet. Now I request Dr. Aniruddha Petkar sir to welcome Chairman of today's valedictory function, our respected Director Dr. Rajendra Satpathy sir by presenting him a bouquet. It's our honor to felicitate you, sir, by a shawl, shreefer and bouquet. Thank you all. Biotechnology is a solution to health challenge, a path to sustainable agriculture, a tool for environmental protection, and a powerful ally of forensic justice. Now I request Dr. Sandeep Sangre sir, Joint Secretary of this conference, to present a brief report of conference proceeding.
Hello, good afternoon all. Respected Director and Chairman of this valedictory function, Dr. Satpathy sir. All the respected dignitaries on the dais and of the dais, all the participants who have came across all over from India, student participants, their mentors, teachers, colleagues, all the teaching staff from my Institute of Science, all the students, I am standing in to present an overview about the today's conference. This one-day national conference, that is recent advances in biotechnology for agriculture, environment and forensic science, acronym as RAB 2026. For this conference, one-day conference, we have received an immense response from all the students all across India. We have received more than 240 registrations, and enthusiastically, we have received total 63 presentations for oral and total 29 poster presentations. Both of these were present online as well as offline. The program started at 10:30, the inauguration session. For this inauguration session, we were fortunate to have the chief guest as Jitendra Kulkarni sir, HOD of Biotechnology Department, Biotechnology, Dharashu Campus, Subcenter, Dr. Babasaheb Ambedkar, Marathwada University. For this, for keynote address, we were blessed with Dr. Anjanaba Bhattacharya, the lead scientist from Maiko Research Institute. We were the chairman for this introductory inauguration session were our beloved director, sir, Satpathy sir. After this inauguration session, we had a keynote address on the topic harnessing induced mutagenesis for next generation crop improvement. So it was very interactive kind of sessions. The science, actually the Bhattacharya sir, he has told about whatever content that we learn in our day-to-day life about mutagenesis, how it is applied in biology or particularly in plant biotechnology. So they were enthusiastic students as well as faculty. They have interacted with them and tried to solve a lot of answers. So he has tried to solve a lot of answers. Even after completing this session, I was witnessing outside the building also they were interacting with him. After this, for the technical session, the second session was conducted by Dr. Khushbu Burat on the topic IPRs as an asset generating tool for biotech and allied fields. After that, we had a lunch, and post lunch, we had two sessions which are run parallel for the oral and poster presentations. As I have told earlier that we have received 63 oral presentation and total 29 poster presentation. So it was a very daunting task for all the jury to screen the candidates with the different topics that they have came up with. After this session, we had the next technical session, which was delivered by Gavle sir, and the topic was application of biotechnology in forensic sciences. After this, we are standing here for the valedictory function, and in this section, after completion or in this session, there will be some prizes that will be announced that will be sponsored by Institute of Biological Sciences. Thank you.
Thank you, sir. I would like to introduce today's chief guest, Shri Narendra Gosavi, sir. Shri Narendra Ramchandra Gosavi, sir, is presently working as a Deputy Director, Directorate of Forensic Science, Maharashtra State, Regional Forensic Science Laboratory, Home Department, Chhatrapati Sambhajinagar. Shri Gosavi, sir, is a senior forensic science professional with over 20 years of experience in forensic investigation and laboratory administration. He completed his MSc in biochemistry from North Maharashtra University in 1998. He began his career as a chemist at Udupharma Laboratories in 1998. Later, worked as a research associate at Dr. Jain Herbal Private Limited from 2001 to 2005. He served as an analyst with the Food and Drug Administration. In 2005, he joined the Directorate of Forensic Science, Maharashtra State. He served as Assistant Chemical Analyst from 2005 to 2011, Assistant Director from 2011 to 2022, as a Deputy Director from 2022 to present. He has published around 20 research papers in the field of forensic chemistry. From August 2022 to August 2025, he handled responsibilities of the DNA and ballistic divisions at the State Forensic Science Laboratory. Shri Gosavi sir is a member of various state-level committees related to DNA and ballistics. He has developed standard operating procedures, SOPs, for the state FSLs, DNA division, and contributed to SOP development for crimes ban operations. He has also served as a member of committee for the Mobile Forensic Ban project under the Government of Maharashtra. He has supervised and contributed to several high-profile investigations, including Kopardi case, Malegaon blast case 2006, chemical factory blast at Manpada, Blue Jet healthcare fire incident at Mahad. And it is very noteworthy that for his outstanding contribution in criminal investigations, he has been appreciated by senior police officials, including the Director General of Police, Legal and Technical, Maharashtra State, and the Commissioner of Police, Mumbai. Sir, your vast experience, visionary leadership, and invaluable contribution to the academic and scientific community inspire us all. May I now request Sri Narendra Goswami, sir, for his address. Please, sir.
Thank you, madam. Respected Chairperson, Dr. Sapute sir, distinguished dignitaries on the dais, eminent scientists, respected faculty members, researchers, and dear students. And ladies and gentlemen, good evening to all of you. It is indeed privilege for me to present here during the function of this national conference on recent advance in biotechnology for health, agriculture, and forensic science, RAP Lab 2026. First of all, I would like to congratulate the Department of Biotechnology, Government Institute of Science, and the Department of Forensic Biology, Government Institute of Forensic Science, Chhatrapati Sambhajinagar.
First of all, I would like to congratulate to Department of Biotechnology, Government Institute of Science, and the Department of Forensic Biology, Government Institute of Forensic Science, Chhatrapati Sambhajinagar, for successfully organizing this national conference. Friends, मी काही शब्द तुम्हाला मराठीत बोलतो. बायोटेक्नॉलॉजी महत्व इतकं आपलं पॉप्युलेशन इतकं वाढते त्या प्रमाणात जमीन दिवसेंदिवस कमी होत जाते. जमीन का कमी होते? इंडस्ट्रियलायझेशन, अर्बनायझेशन याच्यातनं जमीन दिवसेंदिवस कमी होते. तसंच आपण जर बघितलं पेस्टिसाइड इन्सेक्टिसाइडचा वापर सुरू त्याच्यामुळे जमिनीचं नापिकीचं प्रमाण वाढलं. जमिनीचं यामुळे उत्पन्न आता वाढलेलं दिसत असलं तरी आपण नवीन नवीन बियाणांचा वापर करत असलं तरी पुढे भविष्यात हे आपल्याला फूड्स मानवासाठी कमी पडणार आहे. आणि आपल्या पुढच्या गरजा हा बायोटेक्नॉलॉजी विभाग भविष्यात पूर्ण करणार आहे, लक्षात ठेवा. ही जबाबदारी तुमच्यावर आहे. या संशोधनातून याच्यातून तुम्हाला याच्यातून शिकवून द्यायचे आपल्याला भविष्य आपल्या मानव जातीचं अन्न आपल्याला कसं पुरवायचं. तुम्ही सुरुवातच केली वेगवेगळे टिश्यू कल्चर प्रोजेक्ट प्लांट लावून वेगवेगळ्या टिश्यू कल्चरच्या टेक्नॉलॉजीज डेव्हलप करून आपण बरेच आयुर्वेदिक प्लांट टिश्यू कल्चरने वाढवतो. आयुर्वेदिक केळीचे रोपटे सुद्धा आपण टिश्यू कल्चरने वाढवतो. अशा वेगवेगळ्या पुढे याच्यापेक्षा खूप वेगळे करायचे. आणि यात तुम्हाला संशोधनाची दिशा मिळाली पाहिजे. हा शासनाचा सरकारचा आपल्या इथल्या महाविद्यालयाचा हाच उद्देश आहे. जर तुम्ही असं उच्च स्तराचं संशोधन केलं नक्कीच तुमची ओळख जगात वेगळ्या पद्धतीने तुमची ओळख होईल. नक्कीच over the course of this conference we have witnessed excellent scientific discussion, innovative research presentation, and meaningful academic interaction. Such conferences are extremely important because they provide platform for sharing new scientific ideas, presenting research findings, building collaboration between institutions, and encouraging young researchers. Today, biotechnology is playing a crucial role in improving human health, strengthening agriculture, protecting the environment, and supporting sustainable development. At the same time, forensic science has become an essential scientific tool in the criminal justice system, helping investigators establish truth through scientific evidence and such as DNA analysis, toxicology, digital evidence, and crime scene investigation. When biotechnology and forensic science work together, they create powerful opportunities for innovation, research, and social benefit. I am particularly impressed by the energy and dedication shown by young researchers and students during this conference. Your participation reflects the bright future of scientific research in your country. However, you must always remember the science must be guided by ethics, integrity, and responsibility towards society. I would like to sincerely appreciate the organizing committee, faculty members, researchers, and volunteers who have worked tirelessly to make this conference successful. Organizing a national level academic events require vision, coordination, dedication, and the success of this conference clearly reflects those effects. Before concluding, I encourage all participants to carry forward the knowledge gained during this conference and convert it into meaningful research, innovations, and real-world application. Let the ideas discussed here continue to grow through collaboration, publication, and scientific contribution. With this word, I once again congratulate the organizers and participants and with them great success in their future academic and research activities. Thank you very much. Jai.
Thank you, sir, for your motivating and inspiring words. Feedback is a mirror that reflects the success of any academic endeavor. To make this moment memorable, we have selected some participants to share their feedback. So, I now invite Professor Chama Deshpande, Madam, to share her experience about conference. I have called here for giving the feedback of this nice event. Really, this event is very much helpful for the students who are working in the research as well as who are willing to have exposure with this such types of the events also. It definitely helps to build their confidence by presenting their research work in front of the experts. And organization is really nice, madam. And keynote lectures were very nice. Especially I like the lecture regarding IPR and the forensic science, applications of forensic science. It was amazing lecture, sir. And finally, this helps to build the relationship between the institutes among the Chhatrapati Sambhajinagar. Especially, I like this conference and it definitely helps to build the relationship between GIS and MGM because I belong to MGM College of Agriculture Biotechnology, Jandeli, Chhatrapati Sambhajinagar. Thanks for calling me here. I now invite Ms. Arunima to please come forward and share your valuable feedback.
So,
It is on.
hello. So first of all, I would like to thank all of you, like the team members, the management team, the teachers, and the students. It was such a joyful day, and personally, I would like to express my gratitude towards the all members. Like खूप नवीन गोष्टी शिकायला भेटल्या आज आणि अशा काही गोष्टी होते की आपण खूप कमी जे आपले बायोटेकचे आहे जे फॉरेन्सिक सायन्सचे जे नॉलेज आहे ते खूप कमी लोकांना माहिती आहे. तर आज आपल्याला म्हणजे आज जितके सेशन झाले तिथे खूप काही करायला भेटलं. And I would like to thank all of you for all team members and all for organizing these kind of events. Thank you.
Thank you, Arunima. Now we move towards the prize distribution event. These prizes are sponsored by IBS EduTech Chhatrapati Sambhajinagar. I now invite Dr. Pooja Sonone, Madam, for the prize distribution event.
Thank you, Anju ma'am. So I have been given the most important responsibility to declare the prizes for all the efforts that these participants have taken. There are no first, second prizes, so all the presentations are awarded to be the best paper presentation or best poster presentation. So to begin with, for offline oral presentation, I call upon Swadhyay Pitambe to take away his prize of best oral presentation in research scholar category. His presentation was on screening of bacteriocinogenic lactic acid bacteria from floral microbiomes and optimization of bacteriocin production by utilizing statistical productions. Next, I call upon Chaitanya Ingre to take away his award. His presentation was on ecotoxicological assessment of microsynthesized copper nanoparticles on beneficial soil microflora, a step towards informed nano agriculture. From faculty category now, the award for best oral presentation award goes to Anju Pakhale. Her presentation was antibacterial, antifungal and forensic application of green synthesized nanoparticles. Then I call upon Khan Afri Malay Reza from MAT Bhujbal Academy of Science and Commerce. Her presentation was on a comparative study of phytosin nanoparticles and conventional fungicides for disease suppression in Vitis vinifera. Next category is of PG. From PG, I call upon Nakul Kharat from Government Institute of Forensic Science. His presentation was on modified method for enhanced and rapid DNA extraction. Next is Nakul Kharat. Then I also call upon Prachi Goyar from Navy College, Department of Biotechnology. Her presentation was on eco-functional screening of soil actinomycetes for industrial degradation. For offline oral presentation in UG category, I call upon Mirza Anam Riaz Beg from People's College of Forensic Science and Cybersecurity. Her presentation was on age estimation using DNA methylation. I think you must continue clapping. Thank you all. Then in UG category, we have Rutuja Godechor from Devi College. Her presentation was on food adaptation. Then we are heading towards offline poster presentation. The first category is of research student. From research student, I call upon Ms. Shivani Adagare from Maulana Azad College. Her poster presentation was on integrated GCMS and HPTLC based chemical profiling. Is she there? Anyone from her group? From Maulana Azad College, poster presentation. Next, in PG category, I call upon Batise Vinod to take away his Best Poster Presentation Award. His presentation was on green synthesis of phytosin nanoparticles and their application. Then I also call Aishwarya Patwardhan from Vivekanand College. Her poster was on From Weed to Medicine, Biotechnological Formulation and Evolution of Natural Antimicrobial Spray. I also call upon Snehal Bankar from Government Institute of Forensic Science. Her presentation was on deep learning driven STR profile interpretation for automated DNA mixture classification in forensic science. The next category for offline poster presentation is for UG. And from UG, I call upon Chaurasia Surbi from MGM College Agricultural Biotechnology, Gandhili, to take away her award. Her poster title was Extraction of Exopolysaccharides from Lactococcus lactis and Bacillus coagulans by using whey protein. Next is Singh Viran Virender from Government Institute of Forensic Science. His title of presentation was Spot on Safety, a Rapid Milk Adulteration Detection Kit.
Thank you all. Please be seated. With this, we are done with the offline presentation, offline presentation awards. Now it's for online presentation awards. I would be only calling their names. Online oral presentation faculty award goes to Nayana Patil from Jalgaon University. Then Rahul Mahamuni from Dr. Babasaheb Ambedkar Marathwada University. Then from research student, we have Bhavna Suryavanshi from Panve Autonomous School of Commerce and Science. Then Pooja Chauhan, she is from Tamil Nadu, and her presentation was on microbial technology. For online oral presentation, again, PG category, the award goes to Pawar Yesh, and he's from Institute of Science, Mumbai. Then Sunena is from YB Chauhan College of Pharmacy. Online poster presentation. The award goes to Mohini Singh. She is from Directorate of Forensic Science Laboratory, Kalina, Mumbai. Then from PG category, it's Hemant Bhute, and he is from Wardha, Bajaj College of Science. Then Ramni Paugi has again got a Best Oral Presentation Award. She is from Duya College. Then from UG category, we have Asif Pathan from Yashwantrao Chauhan Institute of Science. And the last one is Chauhan Riddhi. She is from Yashwantrao Chauhan Institute of Science, Satara. With this, I declare the awards for the oral presentation, offline, online, and poster presentation, offline and online. I also would thank all the judges for their efforts that they have taken in marking and giving the best efforts and acknowledging them. Thank you very much. Thank you, ma'am. Congratulations to all winners and those who didn't get the prizes. Remember, विद्वत्वत्स नृपत्वत्स नैवतुल्य कदाचन स्वदेश पूज्यते राजा विद्वान सर्वत्र पूज्यते. A king is respected only in his kingdom, but the scholar like you, researcher in this auditorium, is respected everywhere. Now I invite Dr. Mayawati Patil, Madam, convener of this conference, to propose vote of thanks.
Thank you so much, Anju Madam. Once again, very good evening and to all the participants. As now we are moving towards the close-up of this ceremony, so it's my just a duty to propose the vote of thanks to all of one who have directly or indirectly contributed to organize and to successfully conducting these events. So, Honorable Director, sir, respected chief guests, all faculty members and participants. I am just express my sincere gratitude to Dr. Babasaheb Ambedkar Marathwada University for extending the financial support for organization of this conference. I extend my heartfelt thanks to Professor Dr. R. S. Akute, sir, Director, Government Institute of Science and Institute of Forensic Science, for his constant guidance, motivation, and administrative support throughout the planning and execution of this event. Sir, it's your support that motivates us always to engage in such meaningful activities, and we are truly grateful to you, sir, for committed to fostering a vibrant and supportive environment. So, thank you very much, sir. We are deeply honored by the gracious presence and keynote speaker, Dr. Bhattacharya, sir, for sharing his time and valuable knowledge in the inaugural session of this conference. I am thankful to Dr. Jitendra Kulkarni, sir, HOD Biotechnology Department, Sub-Campus, Dr. Babasaheb Ambedkar Marathwada University, Dharashu, for accepting our invitation and for sharing his time with us. A special note of gratitude to Deputy Director, Forensic Laboratory, sir, Shri Narendra Gosavi, sir, for graciously presiding over for today's valedictory session. I sincerely acknowledge the continuous support from the head of the Departments of Forensic Biology, Professor Nimbalkar, sir, as well as head of the biotechnology department, Dr. Harris Panakkal sir, for their leadership and coordination in organization of this conference, for the successfully organizing this conference. My thanks and my appreciation again goes to all the members of the advisory committee, organizing committee, and various subcommittees who have worked tirelessly behind the scenes. Your teamwork and dedication ensured a smooth conduct of this event. Again, a sincere thank to all the judges of online and offline posters and oral presentations who have given their valuable time and expertise to ensure a fair and impartial outcome. Your constructive feedback will surely guide the participants in their future endeavors, sir. I am again thankful to Dr. Pooja Sonone madam, Dr. Manju Pakhale madam for nicely anchoring both of the sessions. We are extremely grateful to our financial sponsors, Alana Group Private Limited and Total Enterprises for their support. Their contribution has significantly strengthened the organization of this conference. We are also express our sincere appreciation towards the our supporting partner, Cellular Edutech Private Limited, for sponsoring and awarding profits to all the winners, which encourage the motivation in our participants. I would like to thank all oral and poster presenters of online as well as offline sessions and attendees from various institutions. Your active participation and intellectual exchange made this conference vibrant and meaningful one. My sincere thanks to all the teachers, teaching staff of both of the institutes for their academic involvement and support, and to all the non-teaching staff again of the both of the institute for their sufficient, efficient administrative support. Then a very big special thanks obviously to our volunteer student participants, without which the organization of this conference was almost impossible. So I think they deserve a huge round of applause for this, whatever the efforts they have taken throughout from the long time. Their dedication, tirelessly effort for managing all the sessions, which include registration, technical arrangement, hospitality, coordination. So all this for that, they are, again truly deserve the appreciation. Again, I would like to acknowledge the team responsible for online streaming and sound mic system for ensuring the comfort and seamless communication to the participants who are joining from the long distance. Due to them, only our online participants could attend every session without any interruption. And finally, I thank each and everyone who present here and contributing for the success of this national conference. Your presence, cooperation, and support have made this academic event memorable and meaningful. So, with this regard, I conclude my few words of vote of thanks. Thank you. Thank you so much.
Thank you, madam. I now request the winners of oral presentation, poster presentation to please come on dais for the photo session. With the permission of chair, I declare that this valedictory function is over. I request everyone to please stand up for the national anthem. I request everyone to please stand up the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you all. Participants are requested to collect their certificates from the registration desk. Thank you all for your participation.
साढ़े सात